Carla, and we are going to present the network model of how the representational structure of a memory is transformed over time and goes from segregation to integration. So briefly, we're going to give a very brief definition of memory from a psychobiological perspective. Memory is the neurocognitive capacity of a living organism to encode, save, and retrieve some information that was obtained during some kind of learning. So in cognitive neuroscience, we work with types of memory. So we have classification for different types of memory. The broad areas are declarative memory and non-declarative memory. And the work that we did here and the paper in which we are based on is inspired in declarative memory, which is basically the type of memory that all humans have regarding the facts and events. And so it's basically being able to retrieve something and put it into words. That's the type of memory that we are interested in. And that has to do with some particular structures and is very highly dependent on the median temporal lobe, specifically the hippocampus. So memories, of course, are encoded in our brain within the neurons. So memories are encoded in groups of neurons that have highly coordinated activity. And declarative memory, particularly in early stages, are very dependent on this structure here, that is the medial temporal lobe or the hippocampus. So memories are also are not static. They can change, and they actually do change over time, and that has to do with changes in the engrams of neurons that are coding that memories. And one way in which we can change or change our memories is by reactivation, which is basically presenting some cue that is related to the original stimuli that encoded that memory, and that creates some changes in the patterns of activity of the neurons that encoded that memory. So, connections between neurons can be strengthened, weakened, new connections can be created, etc. So, something also very important about memories is illustrated in this example that I put here. This is a short story by an Argentinian writer, which is Jorge Luis Borges. And this short story is called Funes el Memorioso. So at some point, the story says, indeed, Funes remembered not only every leaf of every tree of every mountain, but every single time he had perceived or imagined it. And at some other point, it also says, Funes says, my memory, sir, is like a pile of garbage. So this person, Funes, was not able to generalize anything. He could save in his brain every single piece of information he ever perceived. So, of course, this is something that is not very adaptive for biological systems. So we need to have some kind of trade between the persistence of a memory and also the efficacy in which that memory is encoded and saved in our brain. So of course, Borges didn't know much about neuroscience, but these can be related to these models of how our brains encode and save memories that are pattern separation and pattern completion. So this guy Funes, he could pattern separate every single piece of information he ever perceived, and he could not make a generalization or a pattern completion of anything, which is something also very important. And Lucho.
So we can think about different experience in one day of LASCON. For example, an experience of a lecture, an experience of a coffee break, a tutorial, and the experience of a lunch. And these represent the memories of each experience. So for example, this is a community representing the memory of a lecture, the community representing the memory of a coffee break, and so on and so forth. And initially, these communities are poorly connected between each other, so this is a segregated network. But if we reactivate this memory, for example, with another day in LASCON, another lecture, coffee break, tutorial, and a lunch, these communities become more connected between each other. So After several reactivation, this network become more and more integrated. So in a previous work from Babasi and Fuentemilla, the author simulate these changes in the segregation to integration of a network with a simplified model. So they found, well, this represent the degree of integration, and they found that after several reactivation, this network become more and more integrated. And they use a simplified model in which this node could be turned on or turned off, and they use a spreading and plasticity rule. So the main goal of our project is to replicate these results, but modeling this node as a neuron with a more realistic, or indeed a little bit more realistic characteristic. So we use nests with a leaky integrate and fire neurons and 2 plasticity rules. The leaky integrate and fire neuron have all these parameters to be set. We use the standard parameters, and the plasticity use the spike timing dependent plasticity, which strengthens or weakens a pre-existing connection. It strengthens if the presynaptic neuron fires before postsynaptic neurons, and weakens if the opposite happens. And we can set lambda values which scale how much the synaptic weight change. And we also use the calcium-dependent structural plasticity, which creates or removes new connection among neurons. And in this case, each stimulus increases internal calcium level. And if a neuron has have an internal calcium level in this range, this promotes new connection. But if the calcium levels are outside this range, this promotes the removal of a connection. And initially, we start with 4 communities with the same number of neurons each community. And these neurons are highly connected between each other inside the community with a probability of 0.1, but are poorly connected between communities with a probability of 0.0001. So this is a segregate network. And initially, we between 10 and 30 neurons per community were stimulated with a Poisson generator.
Okay, to add to this, the initial weights of these connections were of 1 for external connections and 10 from, I think 10 for internal connections. And the new connections that forms with the structural plasticity rule were with a weight of 1. So this is the scheme that represents the algorithm that we use to generalize the model. First, we took the network that Lucho introduced earlier. Then we set up the nest kernel with the 2 plasticity rules. And then we start a main loop consisting of 10 steps. Every step represents a reactivation. Each step have a Poisson stimulus for 300 milliseconds. Each step of the reactivation lasts for 1000 milliseconds. The first 300, there is a Poisson stimulus. Every 100, the structural plasticity rule updates. And every 10 milliseconds, here it says 100, but it's 10 milliseconds, the plasticity rules and the weights adapt with the STDP plasticity rule. After one reactivation, we extract the values of the weights, the calcium concentration, the degree of integration, the entropy, and the malleability. Here we define the malleability like the relative change of the connections between steps, and H, the entropy of the network. So for the results, here we present the connectivity matrix when each row is a neuron. Maybe we can turn off the light. Well, in the diagonal, we can see clearly the 4 communities. And the outside of the diagonal, we can see that is poorly connected. With each reactivation, not only new connections are formed outside the diagonal, but the weights change for all the network. Then we run 4 simulations for a varying number of neurons, of total neurons, and calculate these parameters that we introduced earlier. When we change this number, the total number of neurons, what we are effectively changing is the effect of the stimulus, because it's fixed at between 10 and 30 neurons for every community that are stimulated. When we have 128, we are stimulating like almost all the community. And we see that we replicate in the majority, for the blue one, the green one, and the yellow one, the monotonous increment that grows asymptotically too. But for the red one, we see that it reaches a maximum and then it starts to decay around the 4 activation. Then to try to interpret that, we see the other graph. For the entropy, we see also this increment monotonically, but for the red one, it starts to decay around the 3rd state, the same as before. The malleability, it decays, it's like the inverse of the entropy, and it starts to converge to 0. And we think that this could be explained because of this graph. This graph represents the calcium mean, the mean level of calcium that the neurons have after the reactivation. That's because it starts in the 1st step. So with every step, it starts to get accumulated calcium, because the tau calcium that we fixed is around 1 second. So there is a remnant after 1 reactivation. And we see that after the 3rd step, it crosses this horizontal line that corresponds to the eta. The eta is the center of the bell that we use to produce or remove connections. In the blue one, it reaches after the 9 reactivation, and in the others, it doesn't reach in this regime. So for the conclusions, we see that in some regime, like in these 10 reactivations, we reproduce the results of the paper for the degree of integration with this network more biological complex. But after, if we let it run, if it starts to like change against the original model. Thank you.
Well, I don't know the original paper, but in comparison to the original paper, what, I mean, advances or what kind of difference, apart from the results, how can you interpret the difference that you obtain in comparison to the original one?
Because in the original paper, the structure is like all the steps, the stimuli and the plasticity are integrated like in a more discrete way. So in the paper, the algorithm, it first apply the stimulus, then apply a diffusion rule, and after the diffusion rule ends in some steady state. Bien. After the diffusion rule reach a steady state, it use the plasticity structural rule. In this code that we use, the two plasticities have an interplay. So that's why we think when we crosses this limit, this threshold, it starts to decrease the level of integration and the entropy.
So your model, in principle, would be better than the other one because you're integrating the two mechanisms. They are acting at the same time. Is that correct?
Using point neurons. Their nodes are not point neurons, right?
No,
we have to in the original model.
In the original model, the neurons are points that could be turned on or turned off, but not.
Yeah, exactly. So binary variables.
Yes,
No
exactly.
dynamics.
Yes, exactly.
The two mechanisms integrate at the same time, structural and synaptic plasticity. Are you able to differentiate the effects of each one in this integration, this segregation to integration transition, to kind of single out which mechanism is the most influential one? No. You didn't do. Yeah.
We didn't show it here because we don't finish analyzing it, but after it crosses this threshold, the average weight of the connection is not here the graph, but in the case, because at the level of the new creation, the new connection are created are more that the synapses are strengthened by the plasticity rule. But when it reaches this threshold, it starts to grow up, and that's why we think that the calcium is, continues increasing. But if you let it ride for like 100 reactivations, the calcium level starts to converge to the equilibrium or to the horizontal line, but from the other side. So this equilibrium is an interplay. I think it's a result of the interplay of these two plasticities.
Okay. Thank you. So the next group, please.
Hi. Hi. We are Anna, Rafael, and Jensen. Today we'll briefly introduce our project entitled "Characterizing Activity Stages." in the Brunel networking using permutation entropy. Traditionally, additive system is described by Boltzmann-Gibbs entropy. However, the brain is not stable, nor does a neural network. The brain is made up of complex system that is sitting on the stage called the edges of chaos that is address of a comprehensive temporal orders and the phase transitions, and thus standard entropy failed to accurately capture the stage. Hence, we're gonna apply the permutation entropy as it better detects the phase transitions that is reflected in the real world neural conditions.
Okay, so we're implementing the Brunel model network. Is a random sparsely connected network that consists of two neuron populations, one for excitatory neurons and one for inhibitory ones. And they have like a small probability of connection between them. In addition, we have like external Poisson neuron population that will generate random inputs to these neurons so they can activate. These neurons, they consist of liquid, integrated, and fire neurons, who each one follow this equation. And the synaptic current in the model is used we're using a current-based approach, where we have like delta pulses when a synapse is when a spike is made. We're implementing the network using the neural simulator to Nest, and yeah, to evaluate the network, we're using two main metrics. The first of them, it measures regularity, which is the coefficient of variation, where when the CV is close to 0, the neuron is spiking in a more regular way, and when it's close to 1, is spiking in a irregular way, like a random spikes. And for the second measure is the phase locking value. That's for the synchrony of the network, where it's given by this equation, where when it's close to 0, the network is in a synchronous state, and when it's close to 1, is in asynchronous state. And to classify the network, we use the following thresholds for each metric. The main point, what we want to investigate is how the network behaves when we change two specific parameters. One is the G, the relative inhibitory synaptic strength, and the second one is the background rate of the network. So this figure is in the Brunel paper, where he classifies the network with these states given this set of parameters in this grid. After that, we want to compare these metrics with the result obtained by using permutation entropy, where we get a time series and we divide it in small segments, and we try to extract patterns from this signal to bin them in these motifs. So on the right figure, we have an example where the first one we have the time series, and the B figure is after calculating these motifs. These are the possible motifs for this configuration. And here we have the histogram, where we can estimate the probability of the signal showing these patterns. So the framework of this project consists in first, we will implement the network and validate the results, try to compare the results of our model with the ones in the literature. After that, we'll simulate the network in the grid parameter space to generate time series for each point. After that, we'll make the complexity analysis using the permutation entropy. And after that, we'll compare the results using entropy with the other two metrics that I showed.
For the results section, we have successfully set up the framework. As you can see on the raster plot on the left, 80% of the neuron populations were excitatory, represented in blue dots, and the rest 20% were inhibitory in red. Then we apply the instantaneous fire rate to the network, represented in distributed activities. At the beginning, there was like a sudden sharp of the network, indicating the network is waking up and reacting to the external drive. Further proving the network is steady and ready for deeper analysis.
Okay, so in this part, we are comparing the individual firing rates of the mean frequency of each population. And the first one, we have like the both populations, the excitatory and the inhibitory. The second one, just the excitatory, and the last one, just the inhibitory population. And with this time series, we can generate these histograms and compare them using statistical tests. So we get this result, and we applying the test, we can verify that all the distributions are equivalent. So from this point on, we're just analyzing the excitatory population for like computational efficiency reasons.
For phase exploration, we focus on two main parameters. The x-axis is conductance inhibition ratio calculated via inhibition over excitation. For the y-axis, the randomized input represents the external drive. Regularity is calculated via CV, and synchrony is calculated via POV. As you can see, as the inhibition ratio goes up, the steady blue zone represents the asynchronous stage.
So here, basically, we're going to compare the all phase diagrams with the original work we're basing. So this is the phase diagram for regularity, synchronous, and you can see there's perfect match. So that's good. And here we doing the classification diagram. So this is from the original work. This is us with our model. And as you can see, the yellow zone is from the SCR state, the blue zone for the AR states, and the green zone for AI states, quite similar. And now, after we validating our model with the another work, we're gonna calculate the permutation entropy. So we calculate the permutation entropy for each point in our grid, change the parameters, and we calculate with 3 values of D, 3, 4, 5, 3, 4, 5. So after this, we fix 3 points to see the activity for entropy. And as you can see here, there's the fixed points in this graphics. So the line blue is represented for the asynchronous states. It is correlated to a maximal entropy. And the green line is for the AI state. It's asynchronous state too, but it's irregular state. So it's quite similar. So this is kind of difficult to us to discern like the difference between the irregular states and regular states. And the yellow line is from the synchronous states. And this states is from the correlation with the low entropy. And that's it.
We have attached the data of cross correlation of seizure symptoms from real-world patients. Traditionally speaking, seizure was considered as a hypersynchrony where the entire brain just fires in a hypersynchronized way. It's either on or off. However, the recent publication indicates a new hypothesis, which is called desynchronization, that the phase of a seizure is made of different phases rather than just on or off. By applying or comparing our computational model with the real-world data, we can easily see that the switch from AR to AI which is from regularity to irregularity, is correspondent with a sudden drop of the real-life cross correlation, which proves that our model validates and potentially support the hypothesis of desynchronization. Also points to the next step research, which is the interaction or comparison between computational model and real-life model of seizure symptoms.
So, to conclusions is we replicated the Brunel network very well using NEST. And about the calculus of entropy, we just can distinguish the synchronous and asynchronous states, and the regular and irregular states is more difficult for us. But I like to say more things about this, like to next steps we can do, because here in the entropy, we just varying the d value, and this entropy we can vary to the tau parameter, is the time lag parameter. And when we change the d value, we just change how much samples we do in each partner. So I think this is important because in our simulation, we just simulation 1000 milliseconds. So this is kind of short to the entropy, and probably it's a hypothesis that's why we can distinguish very well the regular and irregular states. So the next steps I suggest is we just explore more to change the d value, the t value, and see how the entropy activity is gonna work. That's it.
Question?
Just technical question. How much time does it take to calculate the permutation entropy compared to calculate the which is the correlation and the CV basically to get the
Yeah, both the time to calculate both of them was the same. They were about the same time. Yeah. It was just the time like to generate the spiking activity and converting into a signal. Then like to apply the metrics was like the same time.
Sorry, just kind of a silly question. When you show the activity, the time series for each population, are you sure you don't have the same plot for all before that? Yeah, keep going. There, the three plots on the left column. Aren't those the same plots?
No, they are for different populations. Yeah,
Right,
sure.
but they look
Yeah.
exactly the same.
Yeah, the
kind of surprising
distributions
to
are
me.
the same.
Yeah, why?
I try
Yeah, if you
like
can-
I tried different points in different states, and all of them like have the same behavior. I just didn't include them the slides.
So your Poisson input, every single cell receives Poisson
Yeah,
input?
individually is different Poisson input. It's not the same.
Interesting.
Do you have any sort of rationale for the thresholds you use to differentiate between the four regimes, so synchronous regular, asynchronous regular, AI, and AR?
Yeah, so in the work we use as a base to take these threshold values is just because they are the ones that most fit the shapes of the original Brunel model. So if you change that just a little bit, you get like a different phase diagram for the states.
So basically, you're proposing a new method to classify in which of these regimes you are, and as Alain asked, said, Brunel used different measures like the correlation
It's not
length and
very
the variability.
explicit.
And you said that in terms of time, more or less, it takes the same. But would that be any other advantage of your metric in comparison to the one traditionally used by Brunel and other workers?
I think maybe because this one is like more robust, because in the Brunel model wasn't very clear what the criteria he used to divide the states. So this will be like more replicable in other research, I think.
Perhaps something interesting to investigate while you have all the methods into place is the effect of subsampling. Is that, of course, in a Brunel network, you know all the cells because you are simulating them. But now, if you think about applying this to experiments, you will have only access to record only a subset of cells. And then in this case, the question is what is, because of course you can calculate the correlation, you can calculate the CV, and you can calculate the permutation entropy also on a subset of neurons. And that would be interesting to see if this method is more robust, for example, to subsampling. Right? And I think it would be easy to do with what you already have into place.
I agree with you.
So if I understood correctly, these thresholds can only be applied to a Brunel network. If I were using another kind of network, I would have to adjust the parameters accordingly, right?
Yeah, these ones they are set just to reproduce the original phase diagram.
Yeah, but in the case of Brunel, you already know the lines separating. Imagine you're trying to do this in a different network, so you'd have to. What would be the strategy to try and find the thresholds? Say, if you're working on a Potchens-Diesman model or any other network.
I think you have to see how sensible your network will be, like to changes in these parameters, to choose like a suitable threshold value, so like it doesn't like get in the blur situation. Like, I think it's like you have to keep trying and see what fits the best.
Okay. Any other questions?
Okay. So Hello, everyone. I'm González Pelzini, and we'll show you today our computational study on how state-dependent effects of focal stroke lesions may affect whole brain functional connectivity. So the core question that we asked was, does the impact of a focal stroke lesion on whole brain functional connectivity depends on the brain's baseline dynamical state? In other words, if the baseline state of the brain may affect the impact of the same structural stroke lesion. So why it matters? Stroke happens either when there's a block of the blood flow on the brain, which is called ischemic stroke, or due to an hemorrhage when the vessel ruptures. So it's a quite important cause of mortality, the second global cause of mortality, and the third one on cause of disability. And 1 in 4 people will have a stroke in their lifetime, and 53% of strokes occurs in people under 70, while it's more common in people 40 plus years old. On the other hand, depression is a significant risk factor for stroke, because it's also both independent risk factor and a common consequence of stroke. There's an over 50% higher risk of experiencing stroke among individuals with a history of depression. And also, as I said, it's bidirectional, so depression increases stroke risk, and stroke often causes depression. And there's also an overlapping in ages where depression is more common, so there's people between ages 45 to 64 that experience both depression and after that stroke. So we said that stroke lesions can alter functional connectivity well beyond the lesion site, and depression may modulate large-scale network vulnerability. So even if there are papers on post-stroke depression and how depression it's an independent factor risk of stroke, we lack a mechanistic framework for state-dependent vulnerability to these lesions. And so that's why we try to simulate these strokes with a depression baseline. Yeah.
Good afternoon, everyone. My name is Alessandra. So I'm going to explain the methods and the experimental pipeline that we have designed to assess this hypothesis. Since we want to investigate whether the stroke lesion have a larger or a shorter network disruption within the depression baseline than the healthy baseline, we start with two virtual baselines. For the depression, we mainly are going to alter the weights on the nodes that we're going to explain later. We're going to induce 3 focal lesions. We decided to work just with 3, both for the healthy baseline and for the depression-like baseline, from which we're going to obtain using a neural mass model on TBV, simulated both oxygen-dependent signal, and with the functional connectivity and graph metrics, we're going to be able to decide whether there was a disruption index or not. So for the methodology, we're using, as I mentioned, the virtual brain. We're going to work with a structural human connectome. For the parcellation, we have decided to offer the Desikan-Kiliani atlas with 68 regions cortically and 14 subcortical regions. And for the model, we have decided to offer the Hub model. So talking about neural mass models, there are several alternatives. We decided to use the Hub model because it's a model that will allow us to obtain both predicted data. Also, since it's a phenomenological based model, it works with a bifurcation parameter A that if it's negative will be closer to the fixed point, and if it's positive is close to the limit cycle. We remain with the negative part, closer to the fixed point, just to work on. Then talking about the atlas, there are also several options. So we initially we considered the default atlas because there were more regions, like 100, and also there was the AAA atlas from Harvard, I think. But then we faced an issue because in the data, exploring some analyses that were done experimentally before and also simulated, there were some irregularities on whether this increases or decreases the connection between the nodes that we were interested in exploring. So we opt to work with the Desi-Cancliani parcellation. And it's interesting because they experimentally, when the experimenters firstly they did the parcellation, they used 40 MRI scans from patients, and they manually did a segmentation going from sulcus to sulcus and including also the gyrus. We used that with the free surfer software. We also implemented the subcortical regions, which were 14, so 7 per hemisphere. The regions altered for stroke were the rostral middle frontal area, the insula, and the inferior parietal. And for depression, we changed the weights on the rostral middle frontal as well, the rostral anterior cingulate, and the parahippocampal area. Our baseline for the healthy patient or virtual patient will be the resting state functional connectivity in a healthy condition. And for the depression-like, we alter the weights, which were consistent for the regions that we choose within the limbic cortical framework. And those regions and the subcortical regions weren't chosen at random. We based our selections on this paper that correlated the effects that have one region with another. So, yep, that was the base.
So we then decided to decide which lesions we were going to choose. So the middle cerebral artery is the one that supplies blood to the lateral frontal parietal and temporal cortex, as well as deep structures such as basal ganglia, and is also the one that accounts for the majority of ischemic strokes, like 63% of cases. So we decided to focalize the stroke lesions on the frontal, parietal, and also in the insular cortex. Yes.
So moving on to the simulations pipeline. So what we did was generate a volt signal of 1 hour and a half for a total of 3 patients for each lesion for each baseline. So that amounted to a total of like 18 volt signals, which were quite large. And this was quite a challenge to simulate because having building so these long volt signals costs a lot of simulation time. So thanks to Alessandra's laboratory, we could use a dedicated server for this task. And we simulated the volt signals with a sampling of 2 seconds. And we induced a stroke induction after the first 20 minutes. That means that for each lesion, for each baseline, both the healthy and the depressive baseline, we have 20 minutes of healthy signals, and then we input a stroke. And then we see how this effect modulates into the connectivity organization of the functional connectivity matrix. And to do that, we calculated the dynamic functional connectivity in windows of 10 minutes each. So since we have 90 minutes, we will have then 9 functional connectivities separated in 10 minutes each. So the outputs that we got from this are the functional connectivity using the Pearson correlation between the volt signals, and graph metrics from weighted networks, And we also calculated for each of the metrics that were node metrics and not matrix metrics, we calculated a hub disruption index. So we can see how hubs or important nodes are more or less affected than the average nodes. So our results, beginning with the simulation results, we will get one functional connectivity matrix every 10 minutes for each lesion and for each patient and for each baseline. So this is just an example of some of them. Here we can see, for example, a connectivity matrix between a healthy patient and a depressive patient just in the 10 minutes after the stroke. We can see that there's not much difference apart from maybe the brightness of the cells. But even though this difference is quite relatively small, it's kind of organized. So that can translate into differences in the global metrics and the node-specific metrics from the functional connectomes. So we can see the dynamic change of these metrics. So our results will be the whole brain functional connectivities, and we will measure some graph theory metrics such as the mean participation coefficient, the modularity, the mean weight, the global and local efficiency, the between centrality, and lastly, the node strength.
So going, like zooming in the results that we got, first we can talk about the mean participation coefficient to see if these lesion effects depend on the site or not. So for example, we can see here that for the 90 minutes of the simulation, after the onset stroke, there is a decrease on all the baselines, so for the healthy and depressive states. And then rapidly after 10 minutes, it also increases. But we see that for the healthy state, the increase is more regular and more homogeneous, if we can say that. And then for the depressive, there are some regions that are not more homogeneous, like for example the rostral frontal part. So this mean participation coefficient is a network integration metric. So if we have an alteration here, there is a disgregation within the modules. And then we can also see the modularity. And with the modularity also after the stroke onset, there is a rapid decrease and then an increase after 10 minutes. And then we see that it also regulates pretty well here, for example, in the rostral middle frontal part. But for the insula, we see some differences. And this metric of modularity corresponds with the equilibrium between integration and segregation. But we cannot say that there is much difference between the two baselines here. And for the mean functional connectivity weight, we see that this is a more regular metric that tends to increase rapidly and then decrease as well. So we see with three sites that the effects that we have with the stroke have a dependency on the baseline state that we have. We cannot say that based on the effect of the stroke, but we can do say that on the dynamics.
Okay, so moving on, we have also the efficiencies, the global and the local efficiencies, and we can see a similar story as the mean weights of the matrices, that we don't see a clear difference between the baselines for the same lesion. However, between the centrality and the vulnerability rankings, we can see some differences because, for example, here we notice that the healthy patients before the stroke have a pretty stable between centrality, which gives us an idea how the information can pass through some important nodes within the functional connectivity matrix. But our most important results are the hub disruption indexes. A brief explanation of what kappa means. Kappa means it's a measure of how more affected are the nodes that have a high metric. For example, here kappa of the participation coefficient means that if it's negative, it means that the nodes that had originally in the 20 minutes of non-lesion, if they had a high participation coefficient, they lost it. They lost it in a worse way than the average node. And here we can see, for example, that for the between centrality, we have that the depressive have a more positive disruption index, which means that the nodes that have a high between centrality in the baseline healthy portion of the time series remain unaffected or gain more between centrality, which can hint at how the brain reorganizes and makes that nodes that act as the bridges between connections on the functional connectivity behave even more as bridges. So they kind of capitalize the shortcuts between the brain. And we can see that there's no clear distinction here for the kappa of the strength, which is the sum of the weights of each node. So we can see that our main signatures of these differences between baselines can be between the centrality and the participation coefficient.
Okay, so just to summarize, first, lesion effects are clearly state dependent. Second, stroke induces an acute stereotypical network response, followed by partial stabilization. And third, state and lesion site interactions matter. Not all lesions are equally sensitive to baseline differences. And finally, have vulnerability changes with baseline state, supporting a network-informed view of lesion impact. And we want to extend this framework to more locations and severities, longer simulation times. We also plan to test robustness across different atlases and generative models. And the long term is to quantify baseline network vulnerability to help predict the global impact of focal brain lesions. So thank you so much. And we want to acknowledge all the LASCON organizers and instructors and the students.
Would you be able to estimate how dependent is your results on the type of pathological baseline you choose? You chose one, a depression baseline, but if you change it a little bit, would the results be very much different or just more or less similar?
We didn't change the parameters, but we would really like to, in our future directions, to try to see if these different parameters might affect the results. Because, for instance, we saw here that here the rostral middle frontal was also the location of the stroke, and also one of the direct changes that we used to simulate depression. So this was also the one that we observed the most divergent aspect of these coefficients. So this, yes, it would be interesting to simulate other types of depressions based on empirical data. So, yeah.
And also, it will depend on the way of this, how this disease, how this condition, this baseline, manifests in the structural connectome. Because what we did to simulate the depressive baseline was modify the structural connectome according to a paper that we showed, a table that we showed, which showed how the difference between the number of tracts, of white matter tracts, between some regions. There we go. And we can see that they decrease. So we can actually get a number of this. We can say that maybe the connection between these nodes for the depressive case is, for example, an 80% of the control case. So if we wanted to model different conditions, we will need to know if these conditions are structural connectome dependent, or maybe they have more to do with the model itself. For example, we used the Hopf model, and we didn't touch the excitability. I mean, we have a fixed excitability parameter for all nodes, but there are some models for depression, for example, that tune that parameter and not just the structural connectome. So yeah, we will need to explore these other parameters and maybe even incorporate delays in our model and stuff like that.
And as a matter of the baseline, just to complement, we chose 3 regions just for the timing that we have, but we were very interested in taking more hubs to see how these disruptions change, and also based on the stroke post-depression that Gonzalo said, there are some hubs that have another antinode source state, so we like to see if there is any possibility to simulate that with the virtual brain.
Thank you.
So is this the first time you work with those metrics, or were you already familiar with this type of analysis? Oh, it's amazing that you managed to.
Yeah.
Great.
We really like
I would like to make a question mixed with a comment. So you verify that there is a modification in the dynamics depending on the baseline state, okay? But what is the probability of the different dynamics that you see leads to another stroke in another part of the brain? Are you interested in doing something like this? How would you do this?
Like right now, I wouldn't be able to tell you a strategy to do so because to simulate a model, we modified a priori the structural connectome. So we pick a node and we set the tracks, the white matter tracks, all to 0. So we can't. The dynamics is from the functional connectome, and we right now maybe don't haven't explored that strategy to map the dynamics of a functional connectome and see how that affects the structural connectome, because the stroke happens in the structural one. So that's how we model it. So I don't know. The answer is I don't know how I could like modulate the effects from a functional connectome back to the structural connectome. But it's an interesting question.
And that's also something that we were interested on testing at the beginning, but it wasn't like practical on the time we have. But because the fMRI depends on the both oxygen-dependent signals, there is a correlation, right, in the experimental data, that when one region increases the intensity of the signal, then another region decreases. So that's an interesting path that we'd like to explore as well.
About simulating calcium imaging and testing spiking deconvolutions procedures. Okay. All right. So I will start talking a little bit about what is calcium imaging. And it's a technique to visualize and measure variations in calcium within cells. By the technique consists in putting some special fluorescent indicators that lights up when calcium levels rise. And as we know, when a neuron emits action potential, calcium channels opens, which leads to calcium quickly enter the cells, and therefore, by recording these flashes of light related to the calcium imaging, we can see when and where cells are active. And a natural question is how to how to infer the neural now spiking activity from calcium variations. So here is a phrase that Gabriel put here for us. It's like trying to understand the drummer's beat by only looking at the ripples in a glass of water. Okay, and so for this purpose of retrieving the spiking activity for calcium imaging, there's some spiking deconvolution methods that are proposed. There are several tools with this, and some of these are the most used maybe is the OASIS and Lama. The OASIS algorithm assumes that it's based on a simple model in which when a neuron spikes, its calcium, the calcium level increases, and between spikes, the calcium level decreases at a fixed exponential rate. And based on the simple method, statistical inference method is used to give us the probability that each increase in the calcium is in fact a spike or not. And well, one of the criticisms to this method is that it assumes the biology is simple and linear by this exponential decay on the calcium between spikes. And there are still some challenges for calcium imaging, in particular the temporal resolution of calcium imaging that is large, and also that usually some essential neuronal probes as neuropixel or silicon probes are used to confirm the spiking inference on the experiments. So we proposed a new method to spiking the convolution. Well, in words, the idea of the method is simple. The idea is that we simulate, we choose a neural model, we simulate this neural model with different inputs, with different injected currents, and after simulating a lot, we see what is the, well, we take the simulation, we calculate the calcium imaging from the simulated neurons, and after doing it with different inputs, we check what is the calcium, the simulated calcium image that is most similar to the one that we have in a real neuron. So the idea is really simple, and the idea is simple, but to apply this, we need to really to choose exactly what is the neuronal model, how we need to really reproduce the calcium imaging procedure, and this is one the way we did in our project. Is that we choose, we simulate using one of the models provided by et al. It provides us neuronal models for layer 5 pyramidal neurons. It provides 10 neuronal models. For our project, we pick one of these. This is the neuronal model. And so from this neuron, we simulate with the input that we give to this neuron are Poisson stimuli to reproduce the in vivo stimuli. Okay, so as I said, that the idea is simulate with a different of inputs and check what is the input that most resemble the reality. And well, to do this, we use an evolutionary analysis. So we have 10 initial random Poisson stimulus. So we pick the length of the stimulus, the rate of the Poisson point process, and also the strength of the stimulus. These are our parameters that are vary. And using an evolutionary analysis, we compare these simulations with the data. We use the Pearson correlation to check what is the most closest, and by doing this procedure by several generations, we select the simulated data that are closest to the real data. And now Gabriel will talk about the application data.
So for the results, we got here we have the trace, like this is the good. It's a way that is the good because we can visualize the voltage trace here of we generated this red dashed line. So here the dashed line is the calcium simulation created by this input in voltage. And the black one is the real calcium trace recorded from neuron in layer 5 pyramidal cell of auditory cortex of mice. So we can see that like the model it's trying to do the Poisson distribution. Like this is the best correlation that we got for this individual cell, and like it managed to get like one peak at least, and we can visualize that it starts changing there, like how it fires a long time, because the idea is that since we're using a Poisson distribution, we have this different amount, like we randomize the peak amplitude, the frequency of the simulation, and also the peak duration. So this is the result of it. So this is a good result. You can see here, like this, we simulated it for 5,000 milliseconds, which like 5-second recording of calcium image, and like this recording, the whole recording, it were 12 minutes. So like we just got, we extracted a little bit of information because of simulation time. We are also doing it for multiple neurons at a time. So the neurons that I'm going to show, like the one which is the bad one, we got like a really bad like correlation, and can visualize it. But like we see, like it's the different input that were generated to it. So it takes time to the model to converge since it's an evolutionary algorithm. If we increase the amount of population, like the population size of it, we can get a better parameter which can solve it, this neuron. And like we have this situation where it's like, this is the beauty. The beauty because like we can visualize, like the trace can like get at least the format of the calcium trace, and also manage to find some like temporal features of it. And like all of those simulations were done using the same parameters. So we had like 10 population size for 5 generations. So this is super small for an evolutionary algorithm. And like we got like this result. So all of them, it's consistent through with the method. And the proof of concept itself is that we wanted to see, okay, how can we use, like if you apply the OASIS method, which the inference of the spiking, like or spike probability to both methods, the calcium trace, which is simulated, and the real one. So the real one here is the green, and the simulated is the grayish. And when we apply here, we gonna see like this is a different neuron, and this is with a more longer record. So like this is the longest that we managed to get during LASCON, which is 5-second recording. And here we can visualize the probability, like the y-axis here is the probability of the spike. So we can visualize here that we have more fluctuations of it, and we can visualize that like it's similar to what the original method gets, but in our method we can also analyze the spike activity which generated it. So if we got an improved error function for the model, like for the algorithm, and also increase the amount of time for each generation, this can improve even better. So this is just the constraints of the model itself. And if like here just showing like the arrow bar, so all y-axis here are the arrow bar. So if we are trying to minimize the error, right? So we have 5 generations. So here we can visualize that in the x-axis for each plot of this is the different values that we change in Poisson distribution. And we can see that like the algorithm starts like with here with a super bad correlation values, and then it starts optimizing. So each generation you got improving, and like you can visualize that like you got this density of where the parameters of the evolution were going for all the traces that we optimize. It happens for like all 3 different variables that we are changing in our Poisson distribution. So basically that's the idea of the method, and this is how we apply it. It's simple, like it's a reverse engineering problem, but it seems to be reliable with more regenerations. So like we need to take more simulation time to make it better reliable, more reliable. So for the
Thank you, Gabriel. Hello, people. Kadimo already introduced me. My name is Arthur, and I'm here to represent for you our main value propositions. I'm gonna be very simple because we only have like 2 minutes. But basically, what makes our method special is that we can change from a statistical guessing to a biological reconstruction. So basically, it can be more phenomenological in the way we approach the problem and we solve the causal image. So we can swap the engine to study different brain areas. I'm gonna skip this slide very quickly. It's out of order. And another thing about our method is that he's very flexible. So we can easily extend it to networking of spike neurons, more complex and neural models, and other measures to compare a simulation of the data, which different procedures to choose and generate the inputs. And for future work, what we want to do together is that we want to do a systematic comparison with the established deconvolution methods and use datasets with Gauss image and neural pixel recorders in order to provide a grounding truth spiking activity. And in conclusion, basically what we have done in this 1 month in LASCON is basically a way to introduce a flexible and model-based framework for spiking inference, which avoids direct deconvolution and heavily relies on machine learning, and provides an interpretable spiking estimate grounded in neural dynamics. And we don't have a thank you slide, but thank you everyone for the opportunity to be here.
Just a technical question. Imagine I have a super recording with like thousands of neurons by calcium imaging with 2 photons or whatever. Is this applicable in practice to very large data set like that?
Yeah. So yes, like here we did the analysis of like 5 neurons for 2.5 seconds. Like it take around 18 minutes. Like this is because the time of that we wanted to get the results to be done here. So if we like have a good computer to do it, it's you can like increase your amount of time, and you can do it for a bigger resolution. So like for 2 photon calcium imaging, you can do it. And like one thing that we tried to implement it, but it didn't work very well, is that try to create like a video, like the video of like the fluorescence of calcium, and try to apply it to Cayman or like Sweet 2P to see if it's like how it would deconvolve the signal. So
yeah, so this means that if you have many neurons to deconvolve, you could run the procedure once for the whole system,
yeah,
and then apply it to every individual neuron.
Yes.
Is that correct?
That's correct.
Yeah, okay.
You mentioned that you plan to use neuropixels to find. Could you comment a little bit on the strategy that you want?
So the idea of using the neuropixels as a ground truth, or a silicon probe as a ground truth, is to be better to give a better inference to the Poisson stimuli, or the stimuli that you want to generate. Because since you have the spike, you can force like your neuron to spike or the network to spike. So this would be a way of like making the checking the ground truth. This would be really the ground truth. So that's the idea of doing it.
So, do you know which probe was used to get those? Because your temporal resolution seems pretty.
So,
is it GCamp?
Yeah, GCamp 6S.
6, okay. Which
So,
is 40, 50 milliseconds, maybe.
Yeah.
Power?
So there I just since we did this with get like the data from calcium imaging already, deconvoluted. So basically, we just try to match it, and we use the Hill equations. So using the Hill equation, we got the value from GCamp 6F, which were, I think it's 375, and then we need to convert this to the neuron program, which is like it's E minus 6, something like this. And so we applied the equation for it and like ran it. So basically, that were like just a short, like a small temporal, like the amount of frames that we got.
Of course.
Because we could put more frames, but like it took way more time because we were trying to make like 5 neurons at the same time. So
it's kind of impressive that you managed to get
yeah.
the beauty. I mean, the
Yeah.
such
Yeah.
low,
And
severely filtered.
Yeah, and that's the idea of like trying to get like if we can do it for a small time, like we can do it for very amount of time in the small ones, and then do the all trace again. So basically, that's the idea of it for sure.
Okay, I'm sorry for the delay and the confusing. We are Andres, Leonardo, and Matias, and we talk about our project. We try to use a hippocampal model from the C1 layer to and use it for a learning process using a DDM model. But I will explain then. We will talk a little bit about hippocampus, the artificial model, that is the learning model that we used, and phase amplitude coupling. Then we talk about the way that we build the code for this project, a little bit about the result that we achieved, and our comments about this learning process for to code this and the troubles that we find.
Okay, well, thanks. Well, my name is Andres. I will introduce you to this microcircuit. So to get this learning process into a CA1 hippocampal microcircuit, we had, we used this microcircuit published by Ponzi et al. In 2023. This is a very biophysically realistic microcircuit. We have three cell populations. First, we have pyramidal cells with all its complete structure, dendrite, soma, everything. And this is our primary excitatory cell in the model. This cell is connected to two populations that are inhibitory cells. First, we have the PBBC interneurons that are basket cells that they put an inhibitory signal close to the soma of the pyramidal cell. And then we have the OLM inhibitory interneuron that is more like a slow firing interneuron that receives this excitatory signal from the pyramidal cells and send a depressing or inhibitory signal close to the dendrite of the neuron. And this model is in this paper was published with these specifications, but it was really computational expensive. It took in the paper they say that you need a supercomputer to run this. In the same paper they talk about a simplified model with this amount of neurons, and you can see that they erase the population of PBB cells. It's pretty much like this. It's a loop, a feedback between pyramidal cells and OLM interneurons, and we used it. So it's kinda this. So you have the pyramidal neuron that sends an excitatory signal, the OLM receives it and sends an inhibitory signal to the dendrite of the neuron. This is still a very complex system that includes all the channels and every configurations because it's made in neuron and netbind, so every neuron is very complex, and to simulate just 1 second of this in different nucleus of a processor in a computer, it takes like half an hour. So we took that simplified model from the paper and we used it in we had to separate the pyramidal population because to make decisions to learn something we had to we needed two populations, population A, population B, that every population had the same feedback, the same loop with the same OLM interneurons to compare, and every population received that excitatory input directly to the soma, different from the paper that they made the excitatory input in the dendrites. But we still have the OLM population interneurons, two different pyramidal cells populations, and in the paper, this switched by, in the paper they talk about, how can you have the same results without one type of population, and they talk about this decay constant from the GABA A. This constant is the one that allows only the OLM population to regulate this high frequency firing from the pyramidal interneurons. In the paper, they try different values for this decay, and we are using 11.8 microseconds. That is the one that showed more theta gamma phase amplitude coupling that we will talk about it in a minute.
Okay, about the model, a drift vision model, it's a stochastic model that possesses that decision making is not spontaneous, but rather a process of sequential accumulation of noisy evidence over the time. In this opportunity, we use a plausible variant of the model that is called racing model, in which two independent accumulators compete to reach the finish line first. In this model, we have a drift rate that represents the strong evidence for each group, in this case of pyramidal neurons, a leak of decay rate that represents as our classmate explained before, the process of memory loss and the noise that in this model is represent like a Gaussian noise. We try to this model is often used for behavioral data, not for biophysical data. Then we try in this opportunity to need a computer bridge connecting microscale neurophysiological and network activity to a macroscale observable decision-making behavior. For that, we try to implement this racing model with accumulators. Now, the more use A minus V process, but using two accumulators, XA and XV, that represent the both group of pyramidal cells, and the drive rate like synaptic efficacy. We try to simulate a long-term potentiation for the group that has an award each time that the circuit choose this group of cells for threshold, and we use a long-term depression like a punish sequence for the group B, and we work with stochasticity, like use Gaussian noise and a leak too.
Okay, and the phase amplitude coupling, when our project works, is when occurs when the amplitude of high frequency oscillations is modulated by the phase of low frequency rhythm. In our project, this coupling is between theta gamma. This low rating defines time window of rigor and lower stability, controlling when fast oscillations become stronger or weaker. But why this matters? PIC, phase amplitude coupling, enables coordination across temporal scales, supporting information routing, synaptic integration, and cognitive processes such as memory and attention. Behind the formulas, the circuit mechanism, and when PAC occurs, needs a network iteration. It emerges from the feedback loop between pyramidal cells and OLM cells in the CN1 microcircuit. The pyramidal cells and the excitatory neurons in CN1 and generate and propagate gamma activity, and the OLM cells are inhibitory interneurons and they regulate pyramidal cell timing and supporting theta gamma coupling.
Okay, then the way that we use for this project was at first try to replicate, to use the model with the documentation provided by the authors. That was kind of very difficult because the it wasn't well documented and run in all the versions of NetMine and Neuron, and we try to rerun the model, changing something. Then we, in parallel, we try to make a DDM model. At first with a simple X2 scene, simulating an excitatory process, and then we mix the code and use the C input that it's using in the paper. We made an approach that winner take all, and we program LTP and LTD accord to other papers that establish where is a good parameter for that kind of processes. Then we try to restructure the model, changing some parameters and configurations and run and rerun the model, trying to fit in the original model with the process of learning.
So, well, as Matthias said, we started with anything, and I would like to show you one of the first simulations that actually run is this one. So this should be a raster. This is the configuration with the exact same parameters as the paper said for the simplified model. And well, this is because the DTM model had a different input, so we had a completely dead network. After, well, you see the raster like that, the DDM trajectories are meaningful, the choice is 100% bad, the error is really high. But after different configurations, well, we had to change a lot of values in the network, but the principal ones are the conductances between pyramidal and OLM cells, and in the other way, we have to increase it like 60 times for us to see some activation, even also the depression of OLM cells that was really high in the original model, but we had like 34, a value of 34 38 depression, but with a value of 1, we see this activation on, and what that means, that we have a lot of spiking, the model is learning something, but isn't fit anyways. And you can see the history choice that by a very small difference between both of population, the system kind of works, right? So we had to analyze what was happening at another level, right? To see the behavior of the network, and we can see that it has some spiking, it has some activity between theta and gamma, but isn't the best. In the spectrogram, we see some activation near 80 hertz, but it isn't related to the theta phase, because this plot should be like a Gaussian distribution in some kind of way. And after, well, this is one of the latest simulation that we did, and it has better results, I think. You can see between trial 1 and trial 6, there is more inhibition for population B during this 1 second trial. And in time, you see that the model goes up because the weights are really high. You can see them because, well, they switched in for the format, but sorry. And well, you can see that even with this kind of parameters that aren't the best, the accumulation for population A, it's still better than population B, and population B, it's decreasing in time in different trials. And the choice history goes to correct in the majority. In the first trial, it goes bad because there was these values of the weights had create confusion for the system. At the electrical level, you see a better phase amplitude coupling between the gamma amplitude and the theta phase. So you can say that the OLM interneurons are regulating this amplitude in gamma frequencies for the pyramidal neurons. You see LFP activity, and the PSD, well, you can see that theta and gamma are still kind of the same. And well, this simulation is more like, well, we were changing the parameters of the network, but what happens with the parameters of the learning model? So we added some smoothing to this fast increase in the DDM, and we can see that it goes kind of bad. Never goes to the threshold, but even with that kind of bad activity, you have the weights for A increasing, the neural activity for A increasing, and in all the trials, it was the correct decision. And we still have the phase amplitude coupling and the activity between theta and gamma kind of correlated. So, well, to finish this presentation, we will talk about a little bit of the comments. And the first comment is the documentation. We had a lot of trouble with the code, with the actual repository. We found 3 repositories for this paper, and we were reading, but it was a little bit confusing. So if you create some code repositories, please document it well, put the correct comments in English, the values, and that kind of stuff. The next generation will appreciate it.
We find some troubles and this implementation of this code, because the simplified model in the paper is not to be clear about the parameters, and is difficult, so difficult to hard to implementation.
Okay, trying to use a behavioral model to a neurophysiological model, it's a challenge. We didn't find documentation or previous code for that, so it was interesting to do too.
And well, the simulations, if it was for me, right now I will be running a simulation in my computer upstairs, because, well, we did a lot in the last few days, and my computer right now it's burning, but I don't know how much are fine. I don't know if it would, for me, I need the same exact result, so that's the last comment.
Final comment is the code is in GitHub. If anyone wants to run, to send the email to Andres. And thanks Antonio, thanks all the lecturers, all the friends, all the tutors, and we appreciate the time. And thanks, gracias, e one big guest, obrigado.
Let's have 1 minute, but very short question. Any comment? So, what have you learned of your entire work in LASCON?
I think we learned everything, because we didn't know any of this stuff at the beginning. We learned about neurons, these models, to do codes. Neuron and NetPine together for a beginner isn't the best choice. So any LASCONian in the future, don't choose that. Yeah, we had a lot of problem. Yeah. So that's that's what I learned.
I need a better computer.
Me also.
Okay, thank you.
And my group is formed by me, by Amanda and Lucas. And in our project, we have simulated the ping network using an adapted Hodgkin-Huxley model. So okay, so before talking about the analysis and results from our simulation, I'm gonna give you some background on some neurophysiological information, okay? So our brain exhibits several different kinds of oscillations depending on what's going on internally and externally to us, okay? And basically, these oscillations reflect the synchronized activity of a population of neurons in a similar frequency range. Okay, so of particular interest for us here in our project is the gamma range here, which is considered of high frequency. And this is because the neurons, when they start synchronizing their activity in this gamma frequency, they do so in a frequency about 30 or 20 hertz to up to 100 or 150 hertz. So it's not working. Okay. Okay, so yeah. Okay, so now you may be asking yourselves, what's the big deal about this gamma oscillation, right? The thing is, this gamma oscillation is considered not only important but also essential for several cognitive processes. So it's important and interacts tightly with attentional engagement, several types of memory, learning, and sensory perception. And here is just an example of how these gamma oscillations emerge in our brain when primates, specifically rhesus monkey, are performing some attentional task, okay? So what we have here is when a monkey is paying attention to a visual object, or for instance, when you guys are paying attention to what we are saying, what we see here is that the neurons in the visual cortex of the monkeys exhibit a synchronized activity inside this range of the gamma oscillation, okay? So what we are trying to say here and trying to justify our project is that gamma oscillations are important at some level. We don't know the extent of it, but they are important at some level to the healthy or normal functioning of all of these cognitive processes, okay? So it's important for us as neuroscientists, computational neuroscientists, that we better understand this gamma oscillation, how it works, okay? And we know currently that this gamma oscillation at the neuronal level is only possible due to the interaction between excitatory and inhibitory cells. So what we know is the following. This interaction between these two types of neurons happens in this way. And we've seen this at some lectures along this course, right? So the excitatory cell will spike, and it will excite the inhibitory cells, which by hands will suppress the activity of this excitatory cell, and the cycle goes on and on during all of this cognitive process that I've just talked about, okay? So, and is this connection, interaction here between these two types of cells that creates this gamma oscillation. Okay, so this gamma oscillation occurs, as we know, in local populations of neurons scattered across our brain, but it also occurs in different populations of neurons which interact with each other. So, for instance, now, probably, I think, you guys are paying attention on what is going on here. And what is actually happening is that neuropopulations in the parietal cortex, neuropopulations in the prefrontal cortex of all of you guys are interacting with each other. And by this interaction, they synchronize their activity inside this gamma range, okay? So now it comes the most interesting part of our project. One of the several ways of studying and understanding these gamma oscillations is through computational modeling, okay? So several computational models have been created to understand these gamma oscillations. One of the most used and accepted models in literature is the pyramidal interneuronal gamma network, or just PING model. So this PING model simulates the same pattern that we saw between excitatory and inhibitory cells across time. So we have here this interaction, excitatory, inhibitory, and so on. I really don't know how to use that. Okay, so when we apply this model here to a much larger network, we have this same pattern of synchrony. So each dot here represents a spike of a neuron, excitatory in red and inhibitory in blue, okay? So the thing here, and the most important thing for our project, is that this PING network has been implemented using different kinds of neural models, all of them, as far as we know, with successful implementation. However, there are a lot of other neural models that haven't been implemented yet. One of those models are based or are adapted from the Hodgkin-Huxley model. So based on these models that haven't been implemented yet in this PING regime, the objective of our project is to investigate if one of these adapted models from the Hodgkin-Huxley could be implemented in this PING model and show the same pattern of gamma oscillations. So if we can actually show this same pattern of gamma oscillations with this adapted model, which is more, as we already know, more biologically accurate, we could use it to, you know, do several other studies regarding these gamma oscillations. So now I'm gonna pass the word to Amanda.
Thank you. Okay, fine. So as Lon said, we have used a adapted Hodgkin-Huxley model, and it's adapted to cortical neurons. The original one is used for squid, so we had to adapt it to cortical neurons. Okay, and the adaptation made between these two models are, oh my god, what have I done? Are those, and thank you, and our references that we use it to do these changes is that one. Okay. So, talking a little bit more about our specific simulation, we have done two main simulations. One of them has been with only two neurons, one excitatory and one inhibitory. And we did so because we wanted to see if our model reproduces what we expected it to reproduce. Because, as you know, excitatory cells spikes slower than inhibitory cells. But when they are at this region, when they are together, they spike in same frequency. So we reproduce each two cells only to see if it was working, and Lucas will show you after my presentation. And the other simulation we have done has been with a rate of 1 inhibitory cell to 4 excitatory cells, because it is it imitates the cortical context too. And we have used 100 neurons. 80 of them are excitatory and 20 of them inhibitory. And we input a range of current in each of the, only in the excitatory cells. So each excitatory cell receives a different current that is in that range. And these currents are applied in different times also, because we wanted to desynchronize our network, because we wanted to see how it synchronizes with the ping model. Okay. So the things that we have used to analyze our results has been two. One of them has been the PSD, which tells us the dominant oscillatory frequencies of the network and their strength. We used it because we wanted to see if our model reproduces or not what we expected, that was gamma oscillations. And the other thing that we have used is the Kuramoto order parameter, which helps us measure the network synchrony. And as I said before, we used it because we wanted to see how the synchrony changes along the time because of the ping model. And only for you to understand what Lucas will say, the Kuramoto parameter has a range of 0 and 1. And when it's near to 0, you have no synchrony, and near to 1, you have perfect synchrony.
So, talking about the results, the first one is very simple. We have two cells, the excitatory and the inhibitory. The excitatory is the blue trace, and the inhibitory is the red. And we put a current just in the excitatory cell, and for our response to the spike, the inhibitory spike together. So we have this effect for different inputs of current. And when we increase this current, injecting the excitatory neuron, the frequency of spikes increase together. For the network, the first analysis we did, we fixed the probability of connection between the two populations in 50%, and we changed the coupling strength between the two populations. And at first we have this synchronized activity for the excitatory cells, because they receive a stimulus but don't connect with the inhibitory, so don't have any effect in the inhibitory population. But when we increase this coupling, we start to see these dynamics of excitation and inhibition, and in the population that start desynchronized start to have the synchronization after a time of simulation. And when we increase a lot this coupling, emerge these strength dynamics in the excitatory cells, that they are very synchrony because they receive a lot of inputs from the excitatory cells. After that, we fixed the coupling strength in 5 microsiemens, and we changed the probability of connection. So the two first results are very similar to the last ones, but when we put like 100% of probability of connection, so the network is all the connect, we have this strand partner here in the inhibitory population, and high synchronization in the excitatory population. We analyze the frequency of each population. So as we know, the inhibitory cells have a high frequency than the excitatory ones, but we can see like when the frequency in the excitatory starts to increase, we have a response in the inhibitory population. The Kuramoto order parameter at the beginning, when you have this kind of dynamics, the Kuramoto order parameter oscillates, and after a while he starts to increase and it goes close to 1. Here at the beginning we have, in this case, 1 neuron that starts to spike in the beginning of simulation. So in the beginning we have this effect here in the Kuramoto parameter, but in general they characterize the dynamics. And the PSD shows to us that for low gamma frequency we have this preference to the network to have more excited here, and in all the gamma range we have this this capacity, this ability of the network to have more energy and more spikes. As on our conclusions is that the ping model was replicated using this neural model that is more biological, and the network oscillates in the gamma frequency, have a decrease, but in general, we have this effect. The model is dependent of the coupling strength and also the probability of connections between the populations. And if you apply a higher current at the beginning of the simulation, the network tends to synchronize earlier. We want to acknowledge Professor Roque for the organizations and all the other organizers, the lecturers, the monitors, especially Fernando, that helped us with the simulations. And thank you all for paying attention.
Questions? You decided to use a Hodgkin-Huxley type model, which is a biophysically detailed model. Which features of this model would be important for the ping oscillations in this network?
So we test some different features, like different ionic currents at the beginning, but we have some problems with adaptation. And when I try some currents at the beginning, when you have calcium dynamics, the excitatory population tends to synchronize very fast. So you have a very high response in the inhibitory. So we took out this and simulate only the sodium and potassium current, but with this the adaptation for cortical neurons from our work from Tabus Miles. So the difference between the original and this one is some constants and the gate variables of open and closed channels.
And the conductance was important too, because it says about how the excitatory cells are connected to inhibitory cells. So, no, okay. So, yeah, I mean, maybe here. Yeah, when you don't have any connection between the excitatory and inhibitory, you don't generate this ping result, you know. So I think it's the most important. I don't know.
That, I mean, for that you could have just a simplified model.
Yes, exactly.
Did you have a delay between the neuron in the synaptic interaction, or was it instantaneous?
We have a short delay between the synaptic interaction. I think is for inhibitory interactions is around 0.5 maybe. I don't remember exactly. And from excitatory to inhibitory is around 0.1 milliseconds.
this, I guess. So our goal with this was particularly to look at 1F slope and information theoretic measures, specifically the complexity entropy plane, to see are they complementary or redundant when it comes to looking at characterizing brain states. There's all kinds of brain states, being awake, asleep, all the different stages of sleep, being under different levels of sedation. And so we wanted to see, do these add information or are they redundant? In particular, we're interested in reproducing empirical features, empirical feature data with the whole brain model, virtual twin, so that we can understand better what I like to call the virtuous cycle of experimental and computational neuroscience. So, okay, don't need this. It's like too many things in my hand. So we go from having a bunch of data, and from the data, we create models, and from the models, we run simulations to inform future experiments or clinical interventions. So in this case, we're using a whole brain model to understand the effect of varying degrees of propofol in the blood and what that does to brain dynamics and what effect it has on the 1F slope that I showed earlier, as well as the complexity entropy plane. So this will allow us to say, do interventions virtually before we can actually do them in a patient to get a better idea of what it will actually do. And then from there, you can do the experiment, collect more data, refine the models, run more simulations, and hopefully advance your treatment protocols or your experiments that you do in the future.
So yeah, first of all, what we did is look for a dataset that have the conditions that we are interested in to explore. And we find this paper that has an open dataset, is from 20 subjects and 91 channels of eyes closed resting states with different levels of propofol in blood. So here we can see like the typical alpha change in the alpha power when you have more propofol. You can see that the power of alpha, instead of being in the occipital lobe, goes to the frontal lobe. And yeah, so you have the baseline, then you have more propofol, then the moderate, that is like the most propofol in blood in these states, and then the recovery. So this data was like for 5 minutes of resting state with eyes closed. So yeah. Then for the methods that we use to characterize the empirical data, and that we want to also compare to the virtual brain model that we are doing, is with the power spectrum first, that follows a power law. We know that the electrical brain signals like the LFP, EEG, ECG, and even like magnetoencephalography follows this power law, or 1/f to the B exponent, where B is called the spectral exponent or the 1/f slope. And this has been proposed as a marker of arousal and cortical excitation inhibition balance. So here we can see the Gao proposed that when you have like awake states, you have like a flatter slope, that meaning more arousal or higher excitation. And when you have steeper slopes, you have lower arousal and higher inhibition. In this case, it's the same for a dataset of propofol. Here that you can see the same effect, that it has a steeper slope and the baseline has a flatter slope. This is also with rats data that happens the same effect. So yeah, that is what we want to replicate in this characterization of this dataset.
So to get more signatures about experimental data and simulation data, we will use the information theory. That basically is get the right signal, where is have measure to get time series, and we will associate to the time series one PDF. So after that, we can find the entropy and the complex. So the entropy and complex will be used to try to characterize different states of the different brain signal states. So for that, for find the PDF, we use the symbolic methodology of band and prompt, and basically we will have some symbols, and these symbols will be the states of the time series, And basically the PDF will be how 8 symbols appear in this time series. So for that, we have 2 parameters. The first one is the event dimension, D, and it's basically how many points we are used to define the symbol. And then another one is empty time, it's basically which point we are see. For example, if tau is equals 1, we will analyze the neighbor points. But if tau is equal to 2, we are analyze the points with the step of 1. So after they find the PDF, you can calculate the entropy and the complex. So the entropy basically is a mixture of information. So how much information we have of time series, and the complex is, for example, one mixture of symmetry, organization, and etc. So for example, here as an example, for the linear function, we have lower entropy and lower complex. For the noise function, we have high entropy and lower complex. But for the periodic function, it will in the mute of both. So for example, the bright signal is a sum of the periodic signals plus noise signals. So in this case, the brain signal will be in some place like there.
So to actually reproduce the empirical feature data to create virtual EEG data, we used the virtual brain, and we used the Zerlaut model using a data set that is based off of diffusion tensor imaging, and it has 96 different brain regions that are all modeled off of a mean field approximation to adaptive exponential integrate and fire neurons, where you can see the equations down here. So the problem essentially with doing this is that for adaptive exponential integrate and fire neurons, the adaptation term means that it is hard to do an analytical approximation, where when you're doing a mean field approximation, what you're doing is you're replacing the temporal dynamics, the precise things that are going on at the individual spike level, with an instantaneous firing rate. And this is very hard to do in adaptation in the ADX model, because the firing rate actually changes, getting slower and slower as it's getting more stimulation. It doesn't respond to every single input with the same exact output. It adapts. And so in order to get past this, the Zerlaut model actually took a bunch of these ADX integrate and fire neurons, well, more like a single one, and they ran it with a bunch of different parameters to get a statistical model of sort of what would be the firing rate in response to these incoming input currents from excitatory and inhibitory neurons. And using this, they're able to make a semi-analytical solution, which allows you to effectively get a mean field approximation model that allows you to capture the dependence of the firing rate of any particular neuron on the history of spikes that has come into it. And so you see here we have regular spiking neurons, fast spiking neurons corresponding to the excitatory and the inhibitory. Once we have this sort of mean field approximation for individual neurons, bring it into 10,000 excitatory and 10,000 inhibitory neurons, making that into a mean field approximation, because this is how you do it. And then finally, taking these and making these all individual regions inside a brain for a whole brain model. And so for the data set we used, there are 14 subcortical regions, and then 96 minus 82 cortex regions that we're modeling. So these are all modeled actually as kind of cortical columns, and we're modeling the subcortical regions as cortical columns, but it does the job. And so this gives you a signal an external current signal, a simulated signal that's like a local field potential. And this is great, but that's more akin to an IEEG signal, an intracranial an electroencephalogram signal. And so we have to have a way to actually get EEG-looking signals that, you know, actually are comparable to the thing that we see empirically when we do EEG. The best thing about EEG too is that you don't have to cut open someone's brain, and it's very cheap and accessible, and yeah, so it's great if you can get that from your model to make a virtual twin and therefore engage in the virtuous cycle I was talking about previously. The problem, mean field model averages out the kind of necessary data to produce empirically relevant data, and so what we did is added noise ourselves with pink noise. And the assumption being that pink noise captures the way that the local field potential permeates through the skull and captures the non-neuronal sources of noise that give you that nice spectral analysis or spectrum for spectral analysis in the future. Over here on the side, this is a forward projection model that actually details all of the regions in red, and for the black you see all of the channels. And so these are the precise locations of them inside the model and inside the empirical data that we use, where we had 91 channels, 96 regions in red. And so the 91 channels that you can see here are essentially listening to the entire crowd based on the inverse Euclidean distance weighted. So they're not getting the full signal from every single neuron, but a signal that's based on the particular distance that they are from the electrodes. And this gives you that signal that you'd like to have.
So yeah, moving into the results. For the empirical data, we did the 1/f analysis. This is the log-log spectrum of each condition for the 20 subjects. And then we feed it into the 20 and 40 hertz fit, because the data is only from 0.5 to 45 hertz. So we got these slopes. And then we use a toolbox that is called FOOF, and we did a linear fit, and we get this kind of linear fits that are the B exponent that we were seeing after the 1/f or to the B are these exponents of the linear fit that the FOOF algorithm does, and we are replicating the results of Gao and Colombo and a lot of other papers that find that you have flattener slope for the wake or the baseline condition and more steeper slope when you have like more propofol. So we get like the 1/f slope increases with the sedation level and being consistent with the GABAergic modulation of propofol.
So now for the information theory, we choose the MP dimension equals 6 and calculate the entropy and complexity for tau from 0 to 30. So here is for the average of the channels, and we find that the best tau for this data is equal 5. So look for the plan of entropy versus complex. We can characterize the signal. So we see that for more propofol in the blood, we have more entropy and less complex. But in the baseline, that is before to take the propofol, we have more complex and less entropy. So now we can look for all channels, and we can define one distance from one state to the other. For example, here we have the distance, the Euclidean distance between the states and between the baseline and moderate. You can say that this distance is for almost channels. So here we can see the intensity of the distance. So and 2, we can see that the distance is more high in these regions. So now look for the model. We try to apply the same conditions, and we find that for the average of the channels, we have a curve. It's a bit similar to the empirical data, but a little bit different. But we can as well apply the same behavior. So for example, here we have the parameters of the model. It just change these parameters. We can apply this behavior. So here is a compare of the model data and the empirical data.
So yeah, also we did like 1 minute of simulated data, and what we was with the noise parameters on all the default parameters of the Sasha paper. So it's like replicating almost the same model that Alain shows yesterday, and we only changed the tau value of the inhibitory synaptic decays and the adaptation parameter of the excitation of the population. So yeah, for these are the spectrum of the EEG data, and we had different fits, but in the 3 fits that we did, we can find like a similar like qualitatively fits. I mean, it's lower to higher slopes. So also the model data follows the empirical results. So only changing like biophysical parameters, this model is capable of doing like the signatures of true EEG. So yeah. And this also to respond like the question that we did about if 1/f slope and complexity and entropy are saying the same things, we find with a Spearman correlation that is not significantly. And the thing that we did also a mixed effect models, but for the time I will just say that only the condition is what is moving the complexity and the entropy and the slope. So they are saying different things about the brain state. So they are not redundant. It's like a good way to like characterize both of these metrics. So yeah, in summary, the model is capable of to replicate the same signatures of the empirical data, just moving like parameters that are biophysical in space, and here we can see like the same stuff here. And this like also a summary that the 1/f slope systematically increases with propofol sedation, consistent with GABAergic inhibition, and our implementation of the ADEX whole brain model also captures these directions and for the entropy and complexity also. Yeah, and these metrics capture distinct and complementary features of brain states.
Limitations in future directions. So we had quite good fits. It's something like I think 5% off for our complexity entropy plane. I don't think we did a distance as yet to measure how far off our 1F slopes were from the actual empirical data calculated 1F slopes, but I think that we think that we can use simulation-based inference to actually achieve better fits, and that would be good to do for, say, if you're actually trying to make a virtual twin of someone. Well, you take their EEG, you measure the 1/f slope, you measure the complexity entropy of the signal, and then you use simulation-based inference to actually fit the biophysical parameters to them, and then you can use that to make your tests. We also want to tease out what exactly the complementary information that the 1/f slope is telling us. We have some sort of kind of idea about the complexity entropy plane, but with the 1/f slope, there's a lot of contention in the literature over whether it's actually representing just excitatory and inhibitory balance, or if there's something more that it can tell us. And so in this case, it seems like maybe there's something more that it can tell us, and you can use that, both of them together, to characterize different brain states possibly more accurately than you can with just one or the other. We also want to understand why adding pink noise in the way that we did works so well, especially across conditions. We have the baseline, the recovery, the mild, the moderate, and it's the same noise for every single one. And adding that just worked perfectly. No other changes to the model besides tweaking these biophysical parameters of the adaptation and the tau I decay time. So, extending to other brain states would be really awesome, and also exploring topographic differences with our now EEG signal to see the different areas, maybe understand the functional connectivity dynamics, so alpha anterioralization, transient hypofrontality, and things of that nature. These are our references. Thank you!
Thank you. Have time for one very quick. Oh.
Okay, very quick. Nice, but and very quick question is: can you say again at what level you add pink noise?
We add pink noise to the excitatory firing rates that are coming outside of the model, each region. So.
Okay, so you don't put the noise in the equation; you just put the noise up. were solved.
Exactly. So it's
Okay.
like really part of the EEG and not part of the model.
Okay, yes, what I was.
EEG level. It's really, we don't change the model dynamics at all, and it's just the way we're looking at what's coming out of the model.
It's just a quick remark is that exactly what you have done could be done also for psychedelics. Not because the model also exists for psychedelics, but also because there are data in open source of both EEG and fMRI of people under psychedelics. And I think you could just do the very similar analysis. And I don't think they have looked at the 1/a slope, by the way.
Yeah, it was to the output, only the pink noise. Yeah.
Thank you.
Can I start? Oh, hello everyone. My name is Ana, and my group is Emilio and Leo. Our work is about sharp waves ripples. We did an analysis based on article. I will begin the presentation with an introduction to topic. The activity neural study is common level of neural populations, and to it we can use local fields potentials. There are large events, and we can record directly from the local field potentials. And these events, some events are periodic, and it was named abundance frequencies difference. The people use Greek letters to it, like gamma, theta, and the network oscillations represent an hippocampus. And example, waking rat, the local field potential activity of the CA1 region is named theta oscillation, and the 6-10 Hertz range. It is during the preparatory behaviors, such as exploration or some place. But in contrast, during custom behaviors, like immobility, eating, or not sleeping, theta oscillation is replaced by irregularity occurring sharp waves. But what is the sharp waves? there is a right synchronization events and occur in hippocampus. But in our study, we consider the CA3 area in the hippocampus, and we consider because this area is strongly involvement of the generation of sharp wave ribs, and this occur during the offline states of the brain associated this behavior, costumatory behaviors, and non-REM sleep. But it consists a periodic occur of the field potential transient, and a superposed fast periodic oscillation in the range 115 to 150 hertz is derived. And what you can study with it? Because the different stages memories is processing in distinct brain states, and this is can characterization by the oscillator partners, like the sharp waves of the hippocampus. Now I will speak about the basic paper. The article we study, basically we work, is about a network model of area CA3 of a rodent hippocampus. There, they consider 8,000 excitatory pyramidal cells, denominated the place cells, 150 inhibitory basket cells, and all these cells correspond approximately a size of the C area in 600 nanometers thick, like of a hippocampus. In this work, they use to modulate the neurons a single compartment. And the model is adaptive exponential integrate and differential. In these simulations, they split in two parts, corresponding to spatial exploration of the rat on a line. I didn't remember, but is 3 meters, and the offline hippocampus state and slow sleep on the waking mobility. I will show some mathematical tools and they use in the article. This is the model. And this model describes a membrane potential VT and the adaptation variable WT. And here we have some variables like membrane capacitance, and here we have delta T, characterization the sharpness of the thresholds. And in this case, they don't use spikes fixed. They use an exponential function, and to replicate the activity place cells in the CA3 area, they put spike trends during the exploration of the animal and the phase of 2. But they use different rate is lambda I of the place cells, and this is calculation with this equation. And here tau is the spatial the tuning curve, and with it, they use it to learn via STDP. Is is two cases in their study. They use the rule for this model of the spike times depends plasticity to during state offline they use a symmetric rule, and during the exploration they use the asymmetric rule. Is the pattern we use I see in other studies that is more used, and this graph is about the aids about the synapses, and delta t is the difference at time of per-synapse or post-synapse. And here is the equation he was based on that behavior. Here, A is a weight is described the weight update, and it depends of the decayment exponential from the time tau is a constant, and the classical asymmetry what changed the symmetric and the symmetric. A plus is positive and I minus is negative. And the other case, they use the same value to A. And here is some values where here use in the model. Here, ME will show what we did in our work and what we need to rescaling the model.
Okay, the main idea about the author is about the chart wave is to explain the behavior between two main stages. The first one is the online active, is where the animal or mice is walking, and then placer gets fired. The second stage is the offline, where the animal is sleeping, and then some spontaneous activity happen without any external input. But here I will explain briefly and rapidly about the idea of the paper. The first idea is the learning, is where the online stage. It's about where the animal is walking and place cells get fired. They use a spike plasticity learning rule. The decision was because it get easily the connection between the two neurons that get fired together. The second stage is the initiation, is where the happen the chart waves. This spontaneous activity in the paper is a Poisson input that get stronger until exceed a threshold, and when they exceed this threshold, it happens the propagation to the other network, where activates the external connectivity previously mentioned in the paper. And finally, the oscillation. Okay, so what we tried to do in this project, it was firstly try to replicate the result of the paper of Eker. As our second object, it was used, we proposed to use a NetPan and Neuron as a library algorithm because the original code was made in Brian 2. Secondly, it applies a downsampling scaling methodology because the original idea, it was like 8,000 of excitatory place cells, and 150 inhibitory cells. It was computationally cost, and we don't have that resource. And thirdly, our object it was to compare the behavior of this ECTDP rule with the downsampling. And finally, is compare the chart wave behaviors with this downsampling. Okay, so about the methodology, we try to do every step of the author except one. This step is to apply a evolutive algorithm. We can do that, but the other step we apply it. The first one is the same membrane potential exclusively to the pyramid cells, and apply to the same synaptic rule. We downsample. We downsample the computation of the algorithm. We reproduce the activity of the place cells in a linear track. As we get down the size of the place cells, we apply a constant, a DC offset, in order to get similar results, similar behavior. And we, in the stage of line, we apply a Poisson input. And finally, as we downsampling the total size of neurons, we rescale the total synapse scale, and we try to keep the same probability about the pyramid cell connection that is low. Right now, my friend is going to tell about the results.
Thank you. Excuse me, how much time do we have? 5. Okay. Okay, let's go with the results. We did an online model. In this case, we simulate the activation of the place cells during the trajectory of the rat. While it traverses the linear track. So there we can see the activation of the place cells. You can see that there are other pyramidal cells. This is the raster plot of the pyramidal cells. We select the 50% of them to be place cells. And so we also set up a synaptic time dependence plasticity rule. This is a symmetric plasticity rule, and we set up this random weight matrix before running this model with these spikes in the place cells. After that, we obtain this matrix with this weight matrix, and this is similar to the paper, to the result that the paper had. And also we had this replay in the activation of the place cells. It's similar to the paper, but it's different because there is not the sharp wave ripple. So we are going to say, why do we have this result? There is the figure in the paper that shows us how is the sharp wave ripple. There is the sharp wave. It is a wave of 2 hertz frequency, and in the peaks it has like between 150 hertz to 220 hertz of frequency when you do the spectrogram to the local field potential of all of the neurons. We also observe that there is an inverse sequence activation of the place cells here in the raster plot of the pyramidal cells. We didn't obtain that, and we obtained this only one direction sequence of replay. Why didn't we obtain that the same result as the authors? This is because they use a fine-tuning evolutionary algorithm to fine-tune the synaptic weights that involve the inhibitory neurons in that network. Remember that we have pyramidal cells and also basket cells. Those are inhibitory neurons, and we in this synaptic plasticity rule, we only change the synaptic plasticity. I'm sorry, the weight of the synapse of the pyramidal cells to pyramidal cell neurons. So they in the paper they fine tune the other synapse between the pyramidal cells to the basket cells, and that's what is remaining in our project. So we are showing here that it's important to have sharp wave ripples to have sharp wave ripples. It is important to fine tune those inhibitory synapses and also the synapses between pyramidal cells to interneurons. And also we apply this down scaling and down sampling and scaling to our model. It was a factor of 0.2, and we could replicate the synaptic weight matrix that they obtained in the paper with this scaling. So, and also we obtained the replay. So we also apply this adaptive exponential integrate and fire model to our model, but we didn't obtain the sharp wave ripple. So in the paper they say that it is important to apply this model to obtain the sharp wave ripple and also the replay. So here we show that also it is important to fine tune those inhibitory synapses and excitatory synapses to those interneurons. And also, what else can we say? That maybe scaling can preserve average input statistic, but the sharp wave ripples depend on precise interaction between recurrent excitation and fast inhibition. And what are our conclusions? That we could implement this model that was in Brian 2 into a code that implements NetPine and Neuron. And we also apply the downsampling and synaptic scaling from the paper of Romero and et al. Also, scaling preserved learned pyramidal cells to pyramidal cells structure and produce this offline sequential replay that we observed. Despite that, adaptive exponential integrated firing neurons models were applied, we didn't produce the sharp wave ripples. So it's important to apply an evolutionary tuning to those inhibitory synapses. And also the sharp wave ripple generation requires optimized excitation and inhibition balance beyond learning and scaling alone. So thank you all for your attention.
Just a clarification. The model of Ecker et al. Generates a sharp wave ripple, right? But then why do you say it does not generate? Is because when you add STDP, it stops generating sharp waves?
No, we use the same STDP plasticity rule. It was a symmetric one, like this one in the right, but the problem was that we didn't fine tune the synapses between pyramidal cells to basket cells, and also between basket cells and from basket cells to pyramidal cells. That is remaining. They made a fine tuning to those synapses to try to reproduce the sharp wave before running the offline phase. Okay. Thank you.
So hello everyone, my name is Tulio. I'm doing the project with Ayla and Marisol. So I will start with the disclaimer. Marisol couldn't come today because she had a very important meeting with her advisor, but she participated along all the process. So our project, you claim that's the most humble project on the school. So we are trying to reproduce the paper from Vogels and colleagues from 2011. When they implemented inhibitory plasticity to go to excitatory inhibitory balance, and we are using NEST. I will just give a bit of background about the paper. So we discussed it a lot during the school that the cortex operates in the excitatory and inhibitory balance, and but historically, and I will do a lot of connections with the previous presentation. This was defined between equal average amount of depolarization and hyperpolarization currents. And we believe that the brain requires a tighter coupling between this balance to work in time and receive stimulus. And this is, we believe that this is important for a high precision signal processing. So, but we have a problem that is the stability problem. We saw this in the previous presentation, that was not that easy to acquire this excitatory inhibitory balance when you add learning. So the primary motivation of this study was is to work in this condition. And because we know that when you have a task, we have LTP, and the synapses start to become stronger and stronger. And most of the models before this, I just like to highlight that this paper is for 2011. So after this, a lot of stuff came up to it. So before we had a lot of manual fine tuning to maintain the stability. So basically we have excitatory increasing, the inhibition staying quiet. We break this balance and then have an over-excitation and seizures. And I'd like to do advertisement for the podcast of Gaut. I hope that he's watching. We have an episode with Tim Vogels himself explaining not just this paper, but all his work with plasticity. And I particularly like when we see researchers talking about the background of the paper. So we see a big paper, a science paper, very impactful in the field, but it's really interesting to see his perspective and how this hypothesis was generated and how was the process of executing this. So the goal of the paper was to try to tackle this problem of stability, and they searched for a biological mechanism that could allow the brain to self-tune even with a learning process attached to it. So based on this, they hypothesized that we could have a synaptic plasticity in inhibitory synapses that would act like a homeostatic controller. But for this, we should have a rule that could be automatically establish this balance and could drive the network to a synchronous and irregular state. And here we have the equation. This is a more simplified equation than was shown before, but it's the same interpretation. Here we have this target. Firing rate. So if it's above the threshold, the inhibition starts to increase, and if it's below, the inhibition decrease to allow this better tuning. So I'll just do a quick summary of the results of the paper, and then we show what we did. So they are able to show, and then we explore this first figure a lot. They're able to show that this simple rule could drive the network after the simulation with learning to a state that have a lot of excitation and could be balanced, and just this synaptic rule could be balanced. And they also show that if you have a network, this is just one neuron, and they also show that if you have one network, we can drive all these neurons to a synchronous state. We can, here we are using cell assemblies to represent the memory process. And after the stimulus, the network can go back to the synchronous state. And in case of necessity, if you tune this stimulus again, you can recall the cells that were connected. So our project goal, as I say, is super humble. We try to just work to reproduce the first figure of the paper. So we are exploring a lot this first figure, and we try to use NEST. And when we are reading the documentation of NEST, we found that this was already implemented in NEST. So it would be easier for us to use the synaptic plasticity rule. We don't need to go to NEST-ML and do everything by ourselves. But they had a problem that they just have the implementation without any examples and any documentation. So the goal for us was to learn how to use it and to develop a documentation to the NEST. So, and now I will give the floor to Ayla, because I'm not a good guy and she explained the tough part of the project.
So the okay, awesome. So the single neural model was implemented. It was leaky integrate and fire conductance based model, and also to this neuron it was connected 1,000 interneurons. Yes. 1,000, yes, 1,000 neurons, sorry. 1,000 interneurons that is going to be represented by parrot neuron here from NEST. The neurons, the interneurons, were organized in 8 channels. Each channel received 100 excitatory synapses and 25 inhibitory synapses. And each channel was the interneurons from one channel was connected just to one homogeneous Poisson generator, which provides Poisson spike trains in a constant rate, but giving different inputs to the connected nodes. As you can see here, right? Then the inhibitory synapses were implemented using the Vogel-Sperkel synapse rule, which basically uses this learning rate. They put it a very small number to the learning rate, and also this alpha, that is a depression factor, which adjusts the excitatory synapses. And the excitatory synapses was implemented using a static model, so the weight didn't change along the simulation. And for each channel, the excitatory synapse had the same weight. And they calculated the weight from each channel, from the excitatory neurons from each channel, considering the number of the channel. So the synapses with the biggest weight was in the 5th channel. And we were able to successfully reproduce the figure we proposed to do, right? So this is at the beginning of the simulation. The network reverberates just with the excitatory, because they put a very initial weight to the inhibitory synapses. And as the simulate passes, the network is able to regulate itself and to match the current membrane from inhibitory synapses and excitatory synapses in a way that the network achieves an asynchronous irregular state. We also observed that the inhibitory synapses adjusted according to the excitatory synapses with whom they were co-tuned. So the ones that they like from the same channel and also entering in the postsynaptic neuron at the same time, they tend to match, right? The average current from the inhibitory synapse tends to match the excitatory one. And we also tested for different holes. Now and from different holes we achieved to have the same output rate. So the weights were well balanced, and
and the target rate was successfully achieved. The next steps would be to show how the global balance and the detail balance works, and also to implement the Vogler specular rule to a network with 10,000 neurons, right? Matching like the percentage between excitatory and inhibitory. The conclusions.
Just to conclude, so for us the project was a success. So we are able to have a very good experience with Nest. So we managed to use all the ingredients that was already there and put them together to replicate the paper. We have an experience of working on reproducibility, and I have to say that we had an easy life because the paper is very well documented. So it was easy for us to find the parameters, and the only part that was tough, we had Hans at the beginning to help us, so it was smooth this part. And we acquire a lot of skills because we work together to learn how to implement this in Nest. So we spent some time together and alone doing the code and matching how this was working. And when it was not working, we had Pedro to help us. So I can say for us that we had a smooth experience trying to reproduce this paper. And then I would like to thank you all for being here, like in the last day at 5 p.m. So thank you for your attention, and especially thank you, Hawk, for all the effort that he did for us and all this during all the school. So thank you very much.
Questions? How? What would be the? In what conditions would the EI balance break down in your network model? Can you say something about that? How could you?
Based on what we did, with the rule implemented, it never breaks. To be honest, no. I think it would break if we try to implement the learning, and then could be a problem. And we had the problem with other type of neurons when we didn't use the integrate and fire. But this was, if Hans is not watching, this was a problem in the implementation in Nest because some neurons don't accept the negative weights, and then we discussed this a lot because these just work with negative weights. Because at the end, in this equation, they go to the projection from inhibitory to excitatory, and this become negative. And when the Nest doesn't allow it, and then it breaks, so we took a time to realize it, because the initial idea for us was to test with different types of neurons and different type of inputs, Poisson, gamma process, but then we can keep doing this after.
No? If not, thank you.
Because my mom will see me in the YouTube. I'm waiting the time. Yeah. Yeah, 1 minute.
Thank you, Mom.
Okay, hi everyone. Good afternoon. I'm Cynthia Ricaeli. Here is Natalia and Pedro Varela. Today we will talk about the modeling of brain dynamics in patients with primary dysmenorrhea. Okay? So, first the problem. Primary dysmenorrhea is a condition associated abdominal crampings, back pain, and nausea and disease related to menstrual pain. Okay? The prevalence of dysmenorrhea varies between 45 and 95%. So, the areas related to this condition is the sensorio-motor cortex, prefrontal and orbitofrontal, here, thalamus, and limbic system. So, the aim is to stimulate cortical electrical brain signals in women with primary dysmenorrhea using the virtual brain platform and the Jason-Rich neural mass model, and to investigate and better understand the underlying neural mechanisms associated with these conditions. So first, we used the AG real data from 40 women. We processed this data with bandpass filters and removed the artifacts with ICA and extracted the power spectral density and phase locking value only in this band, the theta band. Here is the montage of the AG.
So moving on to our computational and mathematical approach, the model that you use is the Janssen rate model. Unlike the other single neuron models, this is a neural mass model, which means that the model collective activity of large populations of neurons. So the model consists of, we can see here in this diagram, the model consists of pyramidal neurons that receive input from three sources: the excitatory interneuron, that inhibitory interneuron, that both amplify and give an amplified gain and a negative gain on their local input to the local input. So we have this in the semicolon. It's important to note. We have two external inputs from other columns in this model. So we choose this model because it generates EEG-like signals through dynamics of postsynaptic potentials. And we could, with this interchange of these components, we could modulate oscillations, generate EEG signals that I'm going to show you later why this is important for us. So here is the mathematical formulation for the Janssen-Rite model. We have these second order equations here. We have the Y0 and Y3 that model the activities of the pyramidal neurons, and the Y1 and Y4 to the excitatory ensembles, and Y2 and Y5 the inhibitory ensembles. So here we see here a bunch of parameters that we going to need to fit these parameters, and we see here also a function S there is a sigmoid function that transforms the average membrane potential of neurons to the mean firing rate of action potential. Here, for this function, we have the parameters well established in the literature, as noted by Kazemi et al. 2002. We have here. So we use these parameters already fitted, but the other parameters we need to formulate an approach to fit these parameters for our problem, right? So this is how we do it. We choose to do this fitting by using an artificial neural network that I'm going to show later. But for we to train this neural network, we need to first generate a data set similar to ours from EEG data to be able to train this neural network and make prediction later. So we made the following procedure. So we generate 50,000 synthetic EEG channels, EEG signals, for the 32 channels that Cynthia presented at 512 hertz and 1 second each. So the procedures were using the TVB that Natalia is going to explain later. So we vary the ranges of the parameters of Janssen read between this range here, and we generated the EEG signal based on the connective extracted from our data. So we associate these parameters with the EEG data, and we normalize these parameters using linear min-max scaling to serve as labels from the neural network from these nine parameters in the interval of 0 to 1. So we made a procedure that unstable results, any combination of unstable results from these parameters were discarded. So we to validate our model on like real data. Synthetic but approximate to real data, right? So this is the artificial neural network that we use. It's a state-of-the-art network to extract features from EEG data. It's called EEG transformer. It has a convolutional part that consists of convolutional filters that extract scaling and temporal data. So here we have a self-attention as well, self-attention layer that extracts long-range features. And in the end, we adapted here a regression head that uses a fully connected layer and a sigmoid function, and a sequence of 9 outputs that are the Janssen read parameters, already normalized by the sigmoid function. So to train this network, we use the following parameters that is well established in the literature. Use mean squared error that is widely used in problems of regression that we have here. So we use this split of 80, 10, 10% for train validation test. Use 100 epochs and patients of 10. And we save the lowest mean squared error in each epoch. So the final test mean square error was 0.0087. For we see if this error is sufficient for us, is good for us, Natalia is going to compare that EEG generated by these parameters with the real EEG data. So here is the prediction of this trained artificial neural network. We use the real EEG and make a prediction from Janssen read parameters, as we can see here. All these parameters are well ranged between the normal range of standard cases of EEG data, right? As we can see here.
So to integrate everything, we use the Virtual Brain. The Virtual Brain, as we all know, it's an open source computational platform used to simulate large scale brain dynamics. It uses two different types of imaging data. We have structural connectivity data that is from diffusion MRI, and we have functional data, EEG or fMRI. The structural connectivity is basically what it says, explains how each part of the brain is connected in a very simple way of saying it, and the functional data is what we're going to see in the signal, basically. This allow us to do a personalized model of the brain networks, and we especially use the Janssen read model, mainly because in the literature it says that it's very good for not only simulate EEG signals, but also to simulate pain. And in this case, we are simulating pain in, So the workflow basically goes, we already had the EEG process. We had to do a Loretta reconstruction, lead field matrix, and also to set up the anatomy. We did that from an already anatomical atlas that the TBB has. Then we went on to filter everything for the theta band, 4 to 8 hertz, because that is our main interest. And we did the functional connectivity too. From that on, we went on doing the TB brain connectivity. We calculate regions. We calculate where each part of the brain is connected. And we, with what Pedro did, we put those parameters into the Janssen read models so we could get the right oscillations. That is also, yes, the TBB calculates the optimal G, for example. That is one parameter, but for everything else, we had to use what Pedro did. And then we use all of that, all of those files, the lead field, the weight, the length, to simulate the EEG and compare the fit simulated EEG, the standard. The standard is the, the Janssen read model without Pedro's parameters, and the real EEG. And then we just visualize frequency domain and the standard and fitted. Oh, yeah, it's true. So this is the signal that we got. The one in green is a fit model, and the darkest one here is the real data. As you can see, they kind of look the same. It's not perfect, but the oscillations are very good. So what it actually shows is that the standard model alone is not enough. So if we want to have a personalized virtual brain, we have to fit the parameters and we have to extract all the features from a EEG signal. Yeah.
So here we extracted the features, connectivity, functional features, the real data there, and simulated data. How you can see, we have some partners here in these parts. Okay, but in this model, we have a major amplitude. And here we have a video, a short video, showing the connectivity between areas. Display. Here's a, okay. How, okay. This part is the central and parietal areas, and here is prefrontal area. Okay, and this one is the simulate data. Okay, how you can see, have a difference between the connections. We have more connections in the prefrontal area than other, but it's close because we have two connections in the parietal and central area. And we can conclude that the computational modeling reproduces EEG patterns consistent with real data, with particular emphasis on frontal, central, and parietal regions, in agreement with findings from the literature on primary dysmenorrhea and pain processing. These results suggest that involvement of frontoparietal and sensomotor networks and reinforcement the potentials of neuromodules to investigate the cerebral mechanisms underlying menstrual pain. So, thank you guys.
Good questions. You have the Jensen rate model. It has counted 12 parameters. Are you able to, I mean, the number of parameters that you presented there, are you able to pinpoint which parameters are more critical for the identification of this dysmenorrhea pain or not?
Okay. Yeah. The global coupling and the conduction velocity. We change these parameters to do the fitting.
Also, like in most signals too, the A and the B parameters are like the ones that are going to change most of the oscillation. But yeah, the A, the B, and what Cynthia said, I don't.
Yeah, for me, that think more as in the artificial neural network model that I have to generate a bunch of data. I see the C parameters, yeah, has a very large range. So when I generate the parameters, and I generated a C very large, near from 1,350, it's very common that it generates a lot of instability. So we have to generate this data in like an uniform distribution, because like the normal value of C is like 135, is very close to the lower band. So we have to be careful with this parameter as well.
Is it possible to relate some of these parameters to clinical measures so that then people in clinical setting could use your data?
It can useful because we can apply some treatments in the synthetic data and compare, how can I say? Yeah, save time and money, not trying to do experiment, but it simulate this data with some intervention.
I'm sorry. Could you go to the fourth slide before the end? Here. Why you use a different scale for the real and simulated data? You try to use the same scale, because for me I was looking the values, and there will be pretty similar if you use the same scale for both.
No, was proposed because we would like share some partners about our data, because we was looking, oh my God, don't have nothing about. But I chose the highest connectivity to find these partners. So it's proposal to show this scale.
Hey man.
Okay, shall we start or I wait? Okay. Well, hello everyone. Today we are going to present our LASCON project titled Simulation-Based Inference of Neuronal Parameters from Voltage Traces and PRCs. This work has been done by Marcos, Nicolás, and me, Octavio. And the general question to motivate this project is very simple. Can we recover the hidden neural parameters from observable data? So to understand why this problem is important, let's start thinking in experimental points of view. Well, in experiments, what we usually have are voltage traces. And from those voltage traces, we want to infer the biophysical meaningful neural parameters. So here, there may be a problem because different sets of parameters may generate similar spikes in shape and timing. So as you can see in this example, we are showing how the spikes are changing as we change the conductances of the neuron. So there may be a problem here. Why? Maybe the voltage traces are not enough by themselves to a correct estimation of the parameters, because maybe the network that we are using couldn't distinguish very well the set of parameters if the voltage traces that we are using are similar. So we need a way to compare data and see if the data we are comparing is useful to a correct estimation of the parameters. Yeah. So in a few words, the objective is to estimate the neural parameter from a neural model using simulation-based inference, or SBI, and we are going to do it with two types of data. The first one is, once we have a neural model, then we can generate voltage traces, and with these voltage traces we can estimate the parameters. This is the common way. And on the other hand, we can use the voltage traces to estimate the phase response curves of them, which I'm going to explain later, and estimate the parameters. So this methodology gives us an idea to compare both approaches to see which one carries more information. So now to generate the data, we need to pick a model, and we used the Morris-Legard type 2 model. This is a biophysical model based on ionic currents. And you can see in the slide its equations and a simulation where you can follow the system over time in the voltage traces and its representation in the limit cycle, in its attractive limit cycle. And the relevant thing here is you need to know that the voltage traces give us information about this. I mean the system in the limit cycle, not more. So now we can jump to the phase response curves and its definition. The PRCs are defined over a dynamical system with these equations, and they need it needs to have a stable T-periodic solution. That's the limit cycle. And as the dynamics are periodic, we can define a phase to represent where in the limit cycle the system is. Yeah. So when we perturb the system, we are pushing away the system from the limit cycle, and that is represented with an extra term in the dynamical equations. So as we push the system, its phase dynamics changes too. And this change is proportional to the perturbation itself and something, some function of the phase called the phase response curve. This is the definition. So what information the phase response curve gives us? Well, the PRC tells us how the system will relax and come back to the limit cycle, and also how much is the phase shift that the system experiments. So why this could be relevant to estimate the parameters? Why don't just use the voltage traces? Well, because it gives us more information. We can summarize this information in these items, and we can say that the PRC encodes the local geometry in the surrounding of the limit cycle, not only the limit cycle itself, because the voltage traces give us information about the limit cycle, but the PRC just add the information about its surrounding. And it also measures the sensitivity to perturbation. So these two things are information to the system. So we think if we train a network with the PRC instead of the voltage traces, then the network could distinguish the signals even if the same, if different set of parameters makes similar voltage traces. So, now how do we estimate the phase response curve? We use something called the direct impulse method, and it is based in delta function perturbations that makes a phase shift proportional to the PRC. But as we can't compute a delta function in the computer, we compute heaviside functions that represent short current pulses that we apply to the neuron. If we do this in the phase dynamics, we get that the phase shift is also proportional to the phase response curve. So the method consists in, once you have the neuron spiking, you pick a time and you perturb with this pulse, and you measure how the spikes is delayed or advanced. So you can compute this along all the phases in the limit cycle, and you can compute finally the PRC. So here we show an example of how the PRC changes as we change the parameters of the model, just to give you an idea of how it looks like. Well, in summary, we have two amounts of data. One's for the voltage traces, and another one for the PRCs. So the question now is, how do we use them to estimate the neuronal parameters?
Okay, so I'm going to explain the SBI pipeline. This is simulator-based inference, and what we have here is a simulator. Okay, this will be the Maurice-Leclerc, and we fed the simulator with some parameters that we sample from a prior distribution, and we generate the data. Okay, we can generate the spiking or the phase response curves. Okay, and with this data, we fed it into a feature extractor because this is high dimensional, so we need to reduce the dimensionality. And this feature extractor is an encoder. It's parameterized by a multi-layer perceptron, and we get 16 numbers that represents our data. And what we want to do is we want to get this. We want to get the probability of the parameters given the data that the model sees. Okay, and the way that it does this is it samples from a base distribution, usually a Gaussian, which is really simple, and it tries to map this base distribution to the posterior that we want with a function f. Okay, this is what is called flow transformation. And what is important is that this transformation from the base distribution to the posterior that we want is conditioned on these features that are the most relevant features to get the posterior. And what is good about this pipeline is that we can optimize simultaneously the feature extractor to get the most important features of the data, and we can optimize at the same time the flow transformations that goes from this base distribution to the posterior that we want. So, to explain how this flow transformation functions, this is the same. We go to the easy sample distribution, and we try to learn this function that goes to the posterior that we want. And in order to do this, we need a function that is invertible and has a tractable Jacobian just for the training. And this function is actually composed by a lot of G functions that are parameterized with MLPs. So, remember, what we want is the posterior distribution given the data. So, we did a quadratic integrate and fire as a proof of concept to see if the method works. So the integrate and fire method is equation is this one, and we have only one free parameters, and we vary it, and we obtain this kind of voltage traces. And we train with a lot of data the model like this, and then we did inference with some new data, and we look at the posterior distribution that we get, and we see that the model really learned the posterior distribution. You can see here really narrow distributions, all centered on the true parameter. And this is a toy model because this is really easy to infer what's the current if the current only varies the frequency. Okay? So we use this as a toy model, and now we want to go to the Maurice-Leclerc model. And what we want to do is we want to change a lot of parameters and see if the model can recover those parameters just from the data, the voltage time series, or from the phase response curves, and how it differs from one another.
Okay. Yeah. Well, so as my colleagues already said, we use the Morris-Lecar model to simulate different voltage traces and the corresponding PRC. We first change the values of the three conductances to simulate different types of voltage traces and PRC to train the networks. We train two different networks, one with the voltage traces and another one with the PRC, so we can then compare how they both estimate the parameters of the model. Once after training the networks, we simulate testing voltage and PRC. Here we show three different simulations. They are look pretty similar, but are obtained from three different sets of parameters, and we will use this to compare both approaches. Here we show the posterior distribution for the three parameters and the three simulated data. In solid line, there's the posterior distribution for the voltage traces, and in the dashed line is the posterior for the PRC. Well, in this case, we see that both approaches can accurately recover the true parameter value indicated with those vertical lines. And the PRC is a little more precise. We can see that in general the posterior distribution is a bit thinner. So with this, what we did was repeat everything, but now changing 4 parameters: the three conductance, the conductances and the capacitance of the model. And in this case, we found that when we use the PRC to infer the parameters of the model, we can accurately recover the true parameters. But in contrast, when we use the voltage trace alone, we obtain broad distributions, or it's hard to see, but here and here are like almost uniform distributions, meaning that parameter recovery is not possible in these cases. So we try increasing the number of training data from 5,000 to 28,000 and 10,000 for the voltage and PRC respectively. And in this case, we found that for both the voltage and PRC, we can accurately recover the true model parameters. But one important thing about this is that when we use the phase response curve, the parameter estimation is more precise, even though we are using half the number of training data for this. And this means that the estimation based on phase response curves is more robust and efficient for parameter estimation. So to summarize, in this project we use simulation-based inference to recover the true parameters of a Morris-Lecar model. And we found that when we use only the voltage traces, we obtain broader posteriors, especially when we increase the number of estimating parameters. And when we use the phase response curves, we can improve this parameter estimation because of the extra information that these curves provide. In future projects, we would like to use alternative models, for example, Hodgkin-Huxley models. Also apply this methodology to cases in which we have parameter degeneracy, and also test or fine tune this model with some real experimental data. And that's everything. Thank you.
Hey. Okay. Any questions? I'll ask one. I have one question. So great. Is this something you came up with during the school? Right. Was Horacio involved?
Yeah.
Okay.
Yeah.
Right.
Yeah, Horacio was
So
involved.
in the end, as you said, using the PRC is a way of taking transient dynamics into account, right? So would it work if you just used noisy voltage series voltage time series that would have the transient dynamics in them instead of just using steady state oscillations? So if you had noisy data, it would be kicking your traces from the attractor, and you would see transient dynamics as well. And if you train on noisy data, what would happen? And the second question, sorry, is would it work for type 1 excitability as well?
Okay, talking about the first question, I think if you train the data, the network with data with noise, it will depend about which noise you choose, and also voltage traces are like have a lot of points, a lot of data, and the PRC is a way to compact this information. And another pro I see is that the PRC does not depend on the perturbation you apply. So it's a way to you take any perturbation and you already have the information there, and you don't need to think about which perturbation or how much or how strong the perturbation to be applied. I think it could work, but I still thinking that using PRC it will be better because of the amount of data and the type. You need to think about that. Can you repeat the second question?
Stability. Have you tried any model with type 1 excitability?
No, we haven't. But we could. I mean, Maurice Leclerc model, it's also type 1 excitability. So it just change the parameters and run the code and see what we can see. So we can try.
Oh, like this. Okay. I was wondering if it can apply to another protocol, which is something that it would be very useful experimentally. Is that the experimentalists record a neuron, let's suppose, in the visual cortex, and they are interested to know what is the synaptic input to this neuron, without necessarily the neuron spiking. Actually, you can even avoid the neuron to spike by injecting current, and so this would be a pure passive response. The problem is that when you do in vivo recording, very often you cannot repeat. So you need to apply on single trials with no spikes. My question is that, do you think this method could be applied to that case?
I mean, you're talking about having a real neuron?
No, yeah, well, of course you can do you can replicate this with a model, but it's estimating the time course of a synaptic conductance in the membrane, and without necessarily reaching the spiking threshold. Because the voltage is ambiguous about what is the synaptic input. Is there a mixture of excitatory inhibitory input? You cannot know easily, right? But maybe this method can tell. I don't know. That's my question. Yeah.
Maybe there's enough information to estimate the parameters without spiking. Yeah. I don't know. We should test it.
That would be super interesting.
Yeah, also with a type 1 model.
Good, af- Good afternoon, everyone. Our project tell about testing NMDA-dependent synaptic plasticity using Brain2. Our group components by Anahita, Areliane, and Rodrigo. Our project is based in this paper of the Chauvel, Beer, and Cooper. Basically, this paper tells the history of a single synapse, this synapse J, where the calcium entry through NMDA channel and changed the weight. The Chauvel model assumes that calcium is the main signal for plasticity, NMDA receptor are the main source of calcium, and BPAPs have the slow post-depolarization component. In the really, we know calcium isn't the only source of plasticity and the NMDA channel isn't the only source of calcium. We have three hypotheses. First, the main source of calcium is the NMDA receptor, and the calcium dynamics is the difference between calcium entering through the NMDA receptor and how much calcium is being removed over time. Okay, the type of plasticity depends on how much calcium enters the cell. We have two thresholds. The thresholds depression and the threshold potential, where low calcium levels produce no change and moderate levels produce LTD or long-term depression, important for tuning neural circuits, and high levels produce LTP or long-term potential, a process linked to the learning. The third hypothesis is the backpropagation action potential, or BPAP, is the action potential that travels back to the dendrites. And BPAP leaves a slow depolarizing tail that lets the presynaptic neuron know the postsynaptic neuron has already fired. So, different times generate different forms of plasticity. The basic rule of the STDP is if this neuron fires before the post neuron leads to LTP. But, nossa, tá toda errada essa figura. But, if this synapse I activate the post neuron before this synapse J, this connection here is weakened because a neuron forms more than one connection.
So, hi. Can you hear me? Okay. So, now I'm going to talk about our project proposal. So, how Ana says, the model proposed by Chauvel says that plasticity depends on calcium entry through the NMDA receptors. Then, synaptic change should be determined by the NMDA-mediated calcium dynamics. This means that your project is based on this model to test this hypothesis. So this is like this, all wrong, but okay. Now we build a model in Brain2, and we simulate a single synaptic in a postsynaptic neuron where calcium enters through the NMDA receptors, and we determine the synaptic weight. And we also analyze how different stimulation protocols affect levels and the synaptic plasticity. Now, I'm gonna talk about the results, but I want to say that we forgot to put the original graphics to our compare, but when something is wrong that the original, we can say. This first graph is about the BPAP when the BPAP is very short. So, this first graph, we have in this blue curve, we have a NMDA receptor activation.
And in this red curve, we have the postsynaptic despolarization. Here, we have only the pre. In this graph behind this one, we have the calcium in the time. So we noticed that a little bit calcium enters through the NMDA channel. But when we have the post-pre, this means that the postsynaptic neuron fires before the pre. We have this curve in red, but if we compare these two curves, it's almost the same. When we have the pre-pos, this curve it's strong, but it's very brief because there's no have a tail, and calcium enters to NMDA channel a little bit more. But if we remind that Anna say we have LTD and LTP. If these two graphs it's the same, while we can differentiate this to this. There's when we talk about the BPAP if a tail. So it's almost the same graph. If you notice, here we have a tail. Here we don't have. And here we have. So what this means? It's if we analyze only the pre, it's the same. It's the same that this graph. But now we have a tail. The calcium can enter to an NMDA channel a little bit more. And this calcium can cross the depression potential, and we have LTD. And this other case that we have the pre-pos, the calcium cross the potassium threshold, and we have LTP. Okay. Now I'm going to talk about some things. What make the synaptic weights change? So the first is the postsynaptic voltage. Here we talk about the postsynaptic voltage is this red curve. When we have a higher postsynaptic voltage, the higher it's there and lower it's there. So let's explain. This is a fraction. It's the final synaptic weight with the initial synaptic weight. So if it's 1, there's no change in the synapse. So it's the same. But if is writing right there. In this graph, no plasticity was induced below the minus 65 millivolts. But here we have down to this curve, and LTP in this part was induced. So it's between the 60 millivolts, the minus 6 millivolts, and the minus 15.5 millivolts. And if this postsynaptic voltage increase, the despolarization increase, we have LTP. Okay? Now, what happened if we change the frequency? What is this frequency? I think I'm using this board to try and explain. So we have the NMDA channel. So we know that the calcium don't leave the cell through the NMDA channel, but it's just an analogy, okay? If we have one calcium here, this calcium can enter and go out easily because there's no other in this middle. But if we have a lot of calcium, if this one here, if this want to go, it's more difficult to go. This is the frequency. The frequency is the presynaptic neuron fire. So if the presynaptic neuron fires more, we have more frequency. If it fires less, we have a low frequency. It's the same. It's the weight. So Here we have LTD, and if you increase, we have LTP. But here we notice that the graph is continuous because there's a saturation. If the frequency becoming more and more and more higher, the connection between these neurons don't become more stronger. They permanent. They continues the same. And we have this one that it's the weight to the timing between the synapses. And so the pause and the pre. We have a minus because this delta t is the pre minus the pause. So if the pause firing before, if the pre firing before the pause, we have a delta t with a positive number, and the opposite we have a negative. So this part of this graph is the pause-pre, and this part is pre-pause. Here, the same. If we have a long difference between the pause-pre synapse fire, they don't change the connection between these neurons. But if we lower this difference, we have LTD. Now we have an increase because now we have the pre-firing before the pause. And now we have LTD. Here we have again LTP because the time, even if we have the pre-pause, this time it's very long. So the pre-firing very, very before the pause. So this difference is higher, and we have LTD. And now we don't have change to the synapses.
And now I am going to show some results we also replicated from the paper. In this plot, we are observing what is the consequences of manipulating this parameter that determines the tail of the backpropagating action potential. As we saw at the beginning, one of the assumptions of the model is that the backpropagating action potential should have a long tail. This is enough long duration to allow this shape on this protocol, the spike timing dependent plasticity. We see that short periods of time for the back propagating action potential at 15 milliseconds. The window for the induction of the pre-post form of LTD is too narrow, and we see that 15 milliseconds. This is the window for the induction of the pre-post form of the window of the LTD gets wider. So we got better results with the default parameter used in other analysis through the article, the 25 milliseconds. Now we in the next plot we are seeing the results of applying the trace of stimulation of the STDP protocol at different time, at different frequencies. This is STDP protocol consists of trace of stimulations of 1,000 pulses in the from the presynaptic side. So we are seeing that the default parameter of 1 hertz, we are seeing the shape that resembles the STDP form of plasticity. But 10 hertz, we are seeing that it always produces a long-term potentiation, and at 5 hertz, the window for the LTP induction gets wider, and the window for the LTD gets appears sooner. Sorry. And now we are going to observe some results linked to the parameters related to the MDA receptor kinetics. This is important because the plasticity changes as we observe in the development. We link it and changes in this receptor kinetics. We are seeing at the pair pulse protocol of stimulation. We are seeing where the postsynaptic cell neuron is clamped and held at a constant voltage. We see that the window for inducing LTD appears at minor voltages. The less is the value of this parameter represents a slower MDA receptor kinetics. And we see that also the window for in this protocol is applying the pulses of stimulation at different frequencies. We see that at a slower kinetics represented by this parameter, we see that the window for the LTD induction appears at less frequencies, and we see that this also determines the shape of these curves. Also, in this LTD, in the STDP protocol, we see that different MDA receptor kinetics defines the shape of the STDP curves, where the minor a slower value of this parameter, we see there is a larger magnitude on the LTP and a larger and we see that the window where the post pre LTD induction appears later. So as conclusion, we are seeing that this model is important for although it's at a synapse level, we the LTD and the LTD are at the cellular level, the more widely accepted mechanism for learning and injury recovery. We believe that studying even at this the plasticity at this level, at a synapse level, is important though for exploring more complex phenomena like learning and functional healing. So we, um, as we saw in the results, we with that three initial assumptions about the calcium the calcium concentrations as being the important determinant of the plasticity, and the MDA receptor being the main source of calcium entry, and the requirement that states that this the backpropagating action potential needs a long tail. With the model was able to explain these different forms of plasticity induced by these protocols. We are seeing that this experimental we see that this establish a clear link between the MD activation, the calcium influx, and change in synaptic weight. Thank you for your attention. Any question? Okay.
What can you say about whether if one changes the frequency, the stimulation frequency, so you have a dynamic thing instead of just a fixed frequency? If you change it, would that imply some change in your plasticity rule, and some qualitative change in the behavior of the synapse? In other words, if the firing statistics changes over time, so is there any hypothesis?
Okay, we don't look for, but we think this change because the STD, the synaptic weight, have a lot of protocols that change the synaptic weight, but we agree that maybe no. I think that's changed, but we don't see a paper or anything that says that to.
You can use your model to try and
Yes, we
make
can,
some
but
predictions.
we don't try because we finished this weekend, so the time was so short.
Sorry, do you mean inducing plasticity at different frequencies?
You change the statistics of the spike timing. So instead of having, say, a fixed frequency, you change the, say, you have a burst and then say regular one and then fast spiking. I mean, so if you change the statistics of the
Yeah, we
spike
didn't
timing.
try, but we can try that. Yeah.
So next group.
Hello, can you hear me? Okay. Well, hi everyone. We are the final group. We are Lautaro, Marcel, and I'm Pau. And we're gonna talk about psychedelics and how the altered states of consciousness could be something interesting for neuroscience and for health in general. So, give me the next one. Okay. Well, the first that I want to tell you is what is a psychedelic. A psychedelic is a substance, a molecule that is very similar to the serotonin. as you can see, like this is the 3 classical psychedelics. That is the DMT, the psilocybin, the LSD. Here you have the serotonin molecule, and it's pretty similar. So you have like little differences. So, like the simplified idea of this is that these molecules imitates very well the place that serotonin will be in the brain, and that causes like a really hard alteration of consciousness. So the thing is, you have like hallucinations, and you have like a really hard introspective treatment, and things that are really related with therapeutical things. So, but this is also interesting because we can see how the brain behaves in these altered states. So now I'm going to talk about a couple of papers. This is something that is that has been studied for a couple of years, but it's pretty complicated studying this kind of stuff because it's in most of the countries these substances are forbidden. So we have a paper. It's a very recent paper. It's for 2024 that compares the psilocybin with the methylphenidate. The methylphenidate it's a substance that is for ADHD treatment. So the idea of this paper is using fMRI, see if we have some differences between the activation of the regions of the brain using FC matrices. It's a concept that we already saw in the course, but now Lautaro is going to explain it furtherly. The idea is that in this paper they get the result that the psilocybin desynchronizes the brain even more than the methylphenidate, but we will expect that the methylphenidate desynchronizes more the brain. But why this is important? How we can use this kind of stuff? Well, in this paper, they use psilocybin to treat depression, comparing it with escitalopram, that is a very common drug that is used for therapeutical things in depression. And the thing is that in 42 days, they note that the psilocybin was better than the escitalopram. And the idea behind all of this is that with only 1 dose, the psilocybin could be better for depression treatment. But we are not sure about that. We have to study furthermore. So what we're gonna do in this work is to try to see how can we make a model that imitates the brain in an altered state of consciousness. So we're gonna use a model that now we're going to explain in furtherly, that has like little parameters that we're gonna see if they behave different in a psychedelic state.
So to modulate the psychedelic state, we use this model, DMF. This model separates the brain in 90 ROIs, regions of interest, and connect them with this structural matrix. In each ROI, we have 2 neurons, one excitatory and one inhibitory. And the connection between ROIs is given for a connection across the excitatory neurons. So with this model, we could simulate the firing rate across the whole brain, except for the cerebellum. And later we use the hemodynamic model to transform these signals in Bolt signals, the type of signal that we obtain when we measure with fMRI. So this is 2 of the equations of the model. This explain the excitatory firing rate, and this the inhibitory. And I know that it's too late to speak of equations, so don't worry about that. I'm only introduce this for talk about the G, one of the parameters of the model. This parameter control the attachment between ROIs that is modulated by the structural. But with this, we change the intensity of the connection between ROIs. So we have data of fMRI studies of these two works. This both are in the condition of resting state, and this is of DMT, and this of LSD. So in DMT we have 17 subjects and LSD 15. And each subject have data sets from dose and placebo. So for each subject we have this data, I mean a matrix with the 90 ROIs from all the brain except the cerebellum, and the temporal points. We simulate this data like this with the model, so we could compare them. With the bold signals, we construct the FC matrices. The FC matrices are the correlations across all the ROIs. So we obtain something like this. This is a symmetrical matrix because the correlation between the first and the second ROI is equal to the second and the first. And we use this to study if the model is fitting well. This matrix talks about the synchronization across the different ROIs in the brain. So we compute to the FCD. The FCD is kind of similar to the FC, but in this case, we compute we define windows and compute the FC for each window. So we got obtain a lot of FC matrices that will evolution in the time for each window. So for each one of these matrices, we obtain the upper diagonal of the matrix and construct a vector with that we will correlate between all the windows and finally obtain the FCD matrix. So we have the FC and the FC matrices, but how could we say if the fit is good or not? So we define two metrics over these matrices. First is the FC. With the metric of the FC, we will take the upper diagonal of the empirical and the simulated data. And with that, we construct a vector that will correlate, and if the correlation is next to 1, we will say that the simulation is similar to the empirical data. And for the FCD, we again took the upper diagonal of the empirical and the simulated. We construct the distribution of the cells and use the Kolmogorov-Smirnov distance to know the goodness of the fit.
Okay, so a few things to keep in mind. The G is the global coupling, and we have the correlation, which one higher, and the distance, which one smaller. Right here we have simulations for all of the groups, the placebo and those of LSD and of DMT. We can see that the correlation is not highly affected by the variation of G, but the distance between the two FCDs, the simulated and empirical, they are affected by the G, as we can see on these plots that have a minimal, local minima. And then what we saw from this sweep that we did with G is that the dose has a smaller G than the placebo, both on the LSD data and on the DMT data. This is not what we expected. This what the literature shows is that you would have a higher global coupling in the cases of altered states. So yeah, keep in mind that this is a local minima and not global minima. And then what we want to do next is go to another parameter and vary this other parameter. The one that we chose is the JNMDA, which is related with the opening of excitatory channels. And so we chose the LSD dataset because right here we had 30 minutes to do a sweep and 76 minutes to do a sweep for DMT because it's a larger dataset. So we chose only LSD for computational purpose. And right here we have, first of all, we did a preliminary sweep of the values of J and MDA so we could choose them, which was the range that we're going to work with. And this is what we saw from 0.11 to 0.15. And then we did a sweep of varying G as well with different J values. What we can see is that on the correlation, the FC correlation, we have a higher correlation for smaller values of J, both on the placebo and the dose. But when we are looking at the distances on the FD matrices, we have the contrary. We have a lower distance, which is what we want, with a higher J. So here we thought that we need to do a fine-tuning of these parameters because there is like a balance of cost and benefit, a win and loss. So we would have to find the optimal value of J to have an optimal value of G. So it's the best fit for a smaller J. We don't know. We have to do another sweep and probably look at the other parameters of the model so we are sure of how to fit this data and probably analyze the dose and placebo effects. Right? So conclusions. We did not expect having a less G for those, but it probably is a problem because we don't have enough parameters. So it's a local minimum, not a global minimum of the model. We found minimal points of the function G using different values of J. And trying different values of J did not change this tendency of the G, the global coupling, being lower for the dose. So what to do next? Look for other parameters that may influence this altered state and use a computational method to find this global minimum where we could like do a sweep of all of the parameters. And well, this is where we got. Do you want to say something? Okay. Yeah. Thank you.
Questions? What can you say about the brain state? I mean, because you changed it, you analyze the correlations, but what type of behavior would the person be?
Yeah,
depending on the parameter?
we don't mention about that. Like, G is like the global coupling, and the problem that we had in here is like we expect to have more global coupling in the regions when you're in an altered state with psychedelics, because, for example, if you do like this same treatment but with EG, you expect to have like more entropy or like metrics that you expect to be more complex compared to like resting state, you know. So when we saw this problem that with placebo we have like a higher global coupling, we say, okay, we're having a problem, so let's change two parameters at the same time. So we change J and G, because J it's an NMDA receptor, and an NMDA receptor opens the channels that the serotonin enters in the cell, but we don't got to a good fit. So maybe we can expect to use some machine learning methods or things like that, but that will be like more affordable. Yeah. Say something?
That maybe these minimums are local minimums, and this is because in all the space parameters we only are moving in one direction. So if you want to say if this really is a local or a global minimum, we need to add some another directions of the span. So this is the idea of add one another parameter, but is insufficient.
Like it's like two at the end, the exponential end to for that kind of like with 3 parameters. We wanted to do it with 3 parameters, but it will cost like 3 days of computing. So we use 1 and then 2, and that's the time that we have.
Okay, thank you. Any other question?
I like your psychedelic matrices.
Oh, thank you. It's all his fault. Yeah.
So before we leave, just so now I ask everybody but the lecturers and tutors to leave the room, because you have to discuss and come back. Of course, don't go away. You want to know who is the winning, and so we're gonna just discuss what to decide which one is the winning group. So we stay outside and then we'll call you. Sorry? Yeah, okay, but yeah, we'll have, yeah, I know, yeah, you know. think I have to knight you as LASCON. You know, every year, every time there's a LASCON, at the end, I knight the participants, the students, as LASCONians. In general, I give them a certificate of participation. This time, since we're in a digital era, I'm going to send to all of you the certificate with all the specifications so you can come back to your places and prove that you have been here. Okay? So, but I'll do a collective knighthood. May I use? Yeah, but since we don't have a sword, I will use this anyway. Any piece of informatics. So I'll knight all of you, the students, as LASCONians now. So, in the name of Lapik, Hodgkin Huxley, Hebb, Fitzhugh Nagumu, and Roll, I hereby declare you LASCONians. Congratulations to you all. So some final remarks before we go to the last get-together. Okay. So what LASCON is about? I think by now you should know it, but it's probably before. Just now it's oscillating. Yeah. So, well, LASCON, as you know, as I said at the beginning of the school, is an effort to spread and improve computational neuroscience in Latin America and the world, because we have people from the rest of the world here as well. I shouldn't walk. I believe if I walk, it kind of destabilizes it. And also, it attempts to stimulate the habit of using mathematical and computational models to ask questions and seek answers about how the brain works. That's, I mean, I think all of us should agree about that by now. And the strategy of LASCON, you have experienced that, you know this by heart now. Theoretical plus hands-on classes in English. English is, I mean, you have to understand that English, although we love to speak in Portuñol, English is the official language of science. So we have to practice our skills in English as well. Student projects discussed with lecturers and tutors and presented orally, as we just seen today. Different levels and approaches to model the brain. You saw this throughout the school, different approaches, detailed models, simplified ones, very theoretical ones, more high-level ones, depending on the lecture and on the approach. And the faculty made of an international team of leading experts. I think this is a very important topic. I insist on this every time. We invite only the best. I mean, of course, there are other people, but the ones here are among the best researchers in the field. Throughout, I mean, computational science has a very short history, but throughout the history, I think all of them who came to LASCON are arguably the leading lecturers. We have only one here representing, the survival, but all of them, you know. So this is our strategy, you know. But I mean, of course, you came to LASCON because of that, and you know now that this strategy kind of works. But what LASCON is about, besides being just a hands-on computational neuroscience school? Well, it's an opportunity for a student lecturer. I've seen lots of discussions between lecturers and students. Student. It's really pleasant for me to see the students talking to each other during the coffee break and in the parties. So you have this friendship. You establish a network of students which will persist in the future. When you become faculty, you keep this network. And also interactions between lecturers and lecturers. I've seen this during our dinner or at get-togethers in the hotel or even here. People come here and have the opportunity to meet here from different countries and establish and deepen the networking. And in a way that includes not only science but also cultural exchanges. And this is something that I noticed throughout the history of LASCON. I am trying to stimulate it every time there's a LASCON school. I want to stimulate not only scientific exchanges but also cultural ones. We should share experiences from our backgrounds, our cultural backgrounds, because that's the way we can make the world a better place for us, a more peaceful place for us to live. And everything done in the Latin American way. Okay, lots of parties, lots of happiness in our spirit of enjoying life, and of perceiving life or interacting with other people. And now for something completely different. The impact and quality of LASCON should not be judged by the student projects and final presentations, or final presentations. Expecting the students to produce full research results in 4 weeks and explain everything in 20-minute talks is both unrealistic and unfair. So I apologize to all of you for demanding you to do that, because it's really unfair. I mean, 4 weeks is not enough time for you to produce good quality work. Although you produced, but you could produce much better if you had more time. So, but the true measure of LASCON's impact lies in its influence on your future work. So I'll be observing, everybody will be observing you in the future. So in 10 years time, 20 years time, you'll be producing high quality work, I hope, in whatever field you want to choose. Some of you, of course, won't persist in computational science. We will do other stuff, as we have seen on that discussion we had on the first Friday. Some people will work on data science, some people work on different topics, but still, some of the knowledge that you acquired here will stay with you forever, and you'll use this in your life. And this is what matters really for me and the organizing fund agencies, is not the project that you just presented. Some of them could become papers, some of them not, but what really matters is what you're gonna carry with you for the future, for the rest of your lives, okay? And now for something completely different. Time to thank. We wouldn't be here if we're not for those funding agencies like the São Paulo Research Foundation, the International Brain Research Organization, the Neuromathematics Center, the Research, Innovation and Dissemination Center for Neuromathematics, which is this building here, where we are, the University of São Paulo, which is providing us this space, and the Institute of Mathematics and Statistics of the University of São Paulo, which also provided some funding for us. So let's thank all of them. Our lecturers and tutors, of course. So let's thank all of them. Our staff, the two IT guys there, there's a second. And now for something completely different. A final message for young computational neuroscientists like you. The final goal is science. Mastering tools and techniques like those taught at LASCON is fundamental to be a good scientist. But do not mistake them as science. Science is defined by questions, ideas, and understanding. So here you learn a lot of tools. They are essential. Any good scientist should master a couple of tools, a couple of techniques, or more, in order to be able to produce good science. But the final goal of a scientist is to try to understand, try to establish ideas, interpret the data, not only produce data, interpret them, reason about them, and advance the knowledge of people. Okay, so keep this in mind. Don't mistake what you learn here with what you're gonna do in the future, which I hope is true science. And now for something completely different. It's time to announce the winning group of the Best Project Award. And to remember, each member of the winning group will receive a computational neuroscience book of his or her choice. Paperback, preferentially, it's cheaper. So, ta-da-dun, 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 ta-dun. The winning group is... You're all winners. Congratulations to all. But of course, we have to, because I promised a book for the winning group. So we had a tough time to select the winner. So we decided first to attribute some honorable mentions. Some people received this honor. So the honorable mentions go to these five groups here. Pedro Muradas, Florencia Altschuler, who is not here, but hope she's watching us. Karen Cunha. Arthur Pires Bueno, Cadmo Lacha, and Gabriel Queiroz. Federico Smith, Gabriele Müller, and Lindsay Knowles. Giovanna Bortolini, Lucas Doner, and Maria Pérez-de-Oliveira. Nicolás Benner, Octavio Pérez, and Marcos Meo. I hope you come tonight to the get together, because I'll pay to each one of you a caipirinha, or for those who don't drink alcohol, a juice. Okay? Yay! But we needed to select one single group to receive the award. And the winner is, now it's really, I'm going to show it to you. The winning group is Alessandra Latorre, Gonzalo Spezzini, Alonso Canepa. Yeah. Congratulations. So later you send me the books you want. Yeah. We can talk about that later. And before we go to the final party, some remarks on the relationship between computational neuroscience and parties. Parties have had a tremendous influence on computational neuroscience and LASCON. So here's a CNS meeting in Pittsburgh. I was there, but not in that picture. LASCON 2008, LASCON 12. In each LASCON had a big party at the end. You can see some familiar faces here from previous years. So let's keep the tradition. Let's go to the party. But before you go, Boris here has some announcement to give to you. Okay, very briefly. This is Boris Marines.
Who knows? Let's see. Just a second. Tá esperando? Quer usar o computador de vocês? Quer usar? Can I use your computer, Antonio, or yours? Maybe. Sorry about that, guys. Vou tentar aqui, né? Resolução baixou. Eu vou. Acho que tem um dispositivo. Deixa eu tentar. Compatibilidade. Vou pegar ali rapidinho. Sorry about that, guys. So tell it.
I know for something.
Apagou tudo aqui, assim. Tô sem rede, sem nada. Ixi. Deixa eu colocar o web. Agora foi. Tell them a joke or something. Talk about the party or. O que tem que fazer?
O Arthur tá ligando ali, aí vai sincronizar. Aí nós já.
Ok. Ah.
No extended.
Oh, we shouldn't. We're not supposed to see this one. Sorry. Okay. So I told Antonio I would be talking about the next edition of LASCON, but I lied. So out of respect and admiration for you, I'm going to steal your punchline. So. Okay. This is a peteca. I talked to my friends, so apparently in Ireland it's called shuttlecock, which is very weird. And my Spanish-speaking friends said, well, some of them said volante, that would be in Spain, gallo, I think that's in Colombia. I should ask you guys. So this is it's used in traditional Brazilian game. Why not keep the spin in the air using the hands? Oh, microphone, sorry. So allegedly, it was played long before Europeans came to Brazil. Indigenous communities would play this game, and that's kind of true because you can still find indigenous communities playing peteca. And I think every Brazilian child has played peteca at some point in his life. Isn't that true? Oh, there's an exception that proves that all generalizations are wrong. So, right? But the important thing to us is that it gave rise to this expression in Portuguese, which is não deixar a peteca cair. That's the dictionary definition, right? That's, if you can read in your language, we should use more Portuñol and Portuguese and Spanish in LASCON. It's similar to do not drop the ball in English or no bajar la guardia in Spanish. But I mean, not to drop the ball, I imagine a football player carrying the ball, very violent, and no bajar la guardia. Like, no, this is the, well, South American way, but the Brazilian way. So, I mean, it means, literally, do not let the peteca fall, right? But it's used to talk about staying with something and with people, even when things get difficult. It's about paying attention, showing up, and keeping things moving together rather than giving up. In other words, someone who does not let the peteca fall is someone who is there again and again without dropping the ball, or rather without dropping the peteca. So that's exactly what hockey has done with LASCON for the past 22 years. He never lets the peteca fall, even while playing almost every possible role, as you can see in this very nice picture provided by ChatGPT. Designing projects, securing funding, inviting speakers, selecting students, organizing transport, food, and accommodation, picking people up from the airport, driving people around, welcoming everyone. So, and often doing all of this essentially on his own without institutional support, right? So I would like to ask you for your permission to speak on behalf of students, tutors, lecturers, organizers, since I have been in all of these roles at some point, and to offer hockey these small gifts. So. And now for some. Cheers. So and spoiler alert, sorry. Open it, come on. You have to go to the party. Yeah, I wanted to do this before you disappeared into well-deserved rest or wild partying. So, okay, so we prepared this plaque with the names of all students from 2006 to 2026. You can take pictures and translate it, but most of you will understand what This phrase will be understandable to all of you. So there's this neuron-like structure shaped like Latin America. Of course, it looks like a neuron, and its dendrites spread all over the continent, establishing connections. So you can get the reference here. But also, it can also be seen as a tree, a nod to our lush. Oh, well, we still have some lush forests in the continent, depending on the results of our next elections. But it also looks, I mean, a bit like a root or roots, which represents the roots that Antonio has established for us and for the field. Just like in neuroscience, it's all about structure, growth, and connectivity. Oh, sorry. And the next gift I would like to give him is a peteca. It looks a bit like a neuron, doesn't it? Okay, so I'm not sure that we'll be able to play. So yeah, GPT had some trouble. You can see that neural networks still need we need more progress in order to get decent looking hands. Also, since Antonio is from Ribeirão Preto, well, he lives in Ribeirão Preto, he's from Brasilia. I'm from São Paulo, okay. So, I can send you the slides later. We have something which is important to all Ribeirão Pretenses, which is beer. So, I'm not sure you're allowed to drink in here, but you can, yeah. So yeah, the peteca is for you to enjoy in your well-deserved retirement, if you ever retire. So thank you, Antonio, for never letting the peteca fall. Cheers. And so David asked me to project that for you as well. Are you? Would you like to read your poem, David? I need to. Yeah. Where's my? Just the pointer. Is it okay like this, or should I increase the zoom? Are you reading the Portuguese version or the?
No, I can do this.
Okay, sure. So is it okay? Can you see it? Or is it too small? Right. So just if you press page down, you should, no. Try scrolling using this thing.
Hi everyone. After my presentation, I spent some time reflecting, and a few words come to mind about my experiences. I think it may also reflect the experience of the other people here. Maybe it makes more sense in Portuguese than in Spanish or English, but I included the translation anyway. I hope you like it. A cidade que nunca dorme, as pessoas que nunca param, o tempo que nunca voltam e o medo que nunca passa. Passam pessoas, passam lugares, passam momentos, passam cidades. Outro idioma, outros olhares, mesma alegria, mesma saudade. Te vejo hoje, te vejo mais tarde. Quando te vejo, agora quem sabe. Momentos de medo, de alegria, lembro do anseio e da energia, da tristeza do primeiro dia, uma semana de uns 30 dias. Umas aulas eu nem ouvia, mas de verdade, não foi culpa minha. Teve outras que eu só não entendia e algumas eu quase sabia. Ok. Descobri qual é o novo rock, inclusive, obrigado pelo suporte, mas vamos a um bar de menor porte, que tem gente que só quer tomar um porre. Sempre perguntaram se eu tinha perguntas, e eu tinha várias, mas talvez o medo me atrapalhava. Mas eu olhei pro lado e vi que não era só eu o atrapalhado, tinha por volta de uns 60 que também estavam um pouco acuado. Pensando bem, 60 é pouco. Tinha uns 80 meio receoso. Foi bom ter vocês ao lado. Foi ligeiro e demorado. Às vezes parece que passou meio arrastado, mas foi no tempo certo cada passo. Acho que falei mais que devia. Em resumo, hoje chegou ao fim e agradeço a sua companhia. Thank you, everyone.